Good morning, everyone, and welcome to the second day of the WIGGIS Workshop. So again, thanks for being here and attending this event, which has been organized thanks to the support of the LifeU initiative and in particular of the LifeClaw project, of which we are partners, we of the Università Cattolica. Okay, so now I leave the floor to the first speaker of today, Oliver Silmoni, who will talk about a specific type of applications of landscape genomics and similar approaches. But I let the chairman introduce him more properly. Thank you.
Thank you, Licia. Good morning, everybody. This morning, the first speaker will be Oliver Silmoni, as Licia mentioned just before. Oliver is a scientist now at the Carnegie Institution for Science, in both in Baltimore. He's working in. He's also a quantic guy, working at several places, more or less in the same time. So in Baltimore and in the Moy lab in Stanford. But today is with us to present his research in seascape genomics, and he will explain to us how we can use the approaches discussed yesterday in a marine conservation context. So Oliver, the floor is yours.
So thank you, Stefan, and thank you all for being here, and thank you, Licia, for the invitation. So it's a great pleasure to have the opportunity to present some of the things we have been working on with Stefan and with our partners in France, so the partners from the Institute of Research from Development from New Caledonia. So as you can see, we are not talking about livestock anymore. We are moving a bit the focus, and yeah, the methods that we used in these studies are very similar to the one you saw yesterday, but you can see that when we apply landscape genomics to a marine environment, we usually refer to it as seascape genomics. So in this presentation, the idea is to show you a little bit how these methods can be used both to study adaptation of corals to climate change, and at the same time, how can we use this information then to try to protect this environment. So you might wonder why somebody from Switzerland is working on coral reef. And yes, it was, these are a bit the ways of life, but the real reason, or one of the main reasons why anyone should be concerned about coral reef is because coral reef, to put it very simply, are the most biodiverse ecosystem in our ocean. So they cover just 1% of the seafloor, mostly in tropical areas, but they host 1/3 of all the biodiversity in the ocean. So you can see that is an incredible hotspot of biodiversity. So when you look at a reef, you can see a lot of different species. You can see fish, you can see sharks, you can see turtles, crustaceans, and so on. And of course, all these biodiversity richness is then translated in ecosystem services for the human communities. So the livelihoods in the tropics are extremely linked or entangled with the state of the reef. So when you look at the reef, another thing that you notice is that at the center of this ecosystem are hard skeleton corals. And it is very important to understand what corals truly are, because, for example, I started working on this 6 years ago, so at the beginning of my PhD, and I had a really shallow idea of what a coral truly was. So when you look at a reef, you might think that corals are just colored rocks that form a very resistant and resilient structure. But to understand what a coral truly is, you have to look at it from a very close point of view. So this is what we refer to as a coral colony, and you will see in a minute why we call them colonies. But to understand what the coral truly is, we have to have a much closer look. And here we start to see something when you look at a branch of this colony. We start to see this little translucent organism that appears. And if we take a closer look, we see what corals truly are. So corals are animals. They are the same family as jellyfish, but instead of floating around in the water column, they attach to the sea bottom, and they start producing this calcareous skeleton that then constructs the structure of the coral reef. So a colony is basically a colony of polyps that are identical under a genetic point of view. and that together construct this calcareous skeleton that then becomes the backbone or the architecture of the coral reef. And this is the structure where all the fish and all the different animals that live in a coral reef live. So it is important to acknowledge that corals are animal, because once we understand this, we understand also why they are sensitive to the environment. When we have a look at corals here, we can see that there are some little pigments, green pigments inside the flesh of the coral. And these are a bit the reason why corals have been so successful in colonizing the marine environment over the past millions of years. So these are little symbiotic algae that live inside that coral, and they work a little bit like the photovoltaic panel of the coral. So they take sunlight and they transform it in nutrients that are essential for the coral to survive. And this is another important aspect, because if you heard anything about corals on the news over the past year, you heard for sure about coral bleaching. So coral bleaching is a stress reaction that is triggered when coral are exposed to an environmental stress. What we observe at the morphological level is that the coral becomes white. What happens inside the coral is that the coral loses the algae. This symbiosis is broken. And when the symbiosis is broken, coral cannot sustain over long periods. And so if a coral remains bleached over several weeks, it eventually starves and dies. And over the past 30 years, we have been observing more and more recurrently coral bleaching phenomenon occur at massive scale worldwide. So they are mostly associated with anomalous changes in water temperature. And as I said, they're becoming more and more frequent. Here in this picture, you can see what a coral reef looked like before a bleaching event. Then we have the heat waves that arrives, and all the coral reef all of a sudden becomes white. This state persists over 1-2 weeks, and after that, the entire coral of the reef die, and the seaweed and macroalgae overgrow the coral reef, destroying the three-dimensional structure of the reef. And by 2050, we expect that these bleaching conditions will become persistent worldwide. So the thermal conditions that cause coral bleaching are expected to become persistent at worldwide scale in 30 years from now. So the question now is, are we in an hopeless situation for a coral reef and for biodiversity in our ocean? And the truth is we might be in an hopeless situation, but over the past decades, we have been observing more and more research reports as this one here. So there are some coral reefs that have been exposed recurrently to thermal stress over their history, and corals at this reef show an increased resistance to thermal stress. So these studies are based on a local observation. So we have the feeling that there are some coral reefs, this is not the majority, but are some reefs that might be adapting to climate change. The corals there might have been developing genetic characteristics that allow them to be resistant to climate change. The problem is, how can we find this coral? How can we find it across a spatial scale that makes sense then to empower conservation strategy and know where to set up conservation priorities, for example. And you can see this very clearly when we look at one of the largest reef system in the world, which is the Australian Great Barrier Reef. So here in the northwest of Australia. So all the green area that you see in this map are coral reef. The Great Barrier Reef is here, and the observation that we have on where these adapted populations are so far is very precise. As you can see here, we can have really local information on the adapted or non-adapted reef. The problem is that this area is as big as Italy, and we cannot base a conservation strategy just on information that we see at a very tiny location compared to the size of the Great Barrier Reef, because the truth is there could be thermally adapted corals a bit everywhere in Australia and as well in the rest of the world in the neighboring reef. So what we did in our thesis was to try to fill this gap in coral conservation and in the studies of coral adaptation. The first thing we wanted to, the question we wanted to answer is whether, how can we find adapted population of corals? And if we manage to find how can we use this information to empower the conservation of coral reef. And we did it using this landscape genomics applied to the marine environment, so seascape genomics. So I won't be long about the methods because yesterday Elia already showed us what the seascape genomics or the landscape genomic method is, but just to remind you, it's a method to study adaptation in the field, combining environmental analysis from one side and population genomics on the other side. So the idea behind the analysis that we wanted to perform is to look at the coral reef and start using some satellite imagery. So we have satellites that have been up in the air for 40, 50 years now, and they are measuring water temperature over the past decades. And then using this information, we can characterize what water temperature has been like across the reef worldwide over the past years. Based on this, we can select some reef that have been exposed to high or stressful environmental conditions and other that have not been exposed to thermal stress. We go to this reef, we scuba dive, we collect coral samples, and we run a genetic analysis. And the goal of this genetic analysis is to uncover what are the genetic characteristics that make corals resistant to temperature. So in short, as we saw yesterday, the idea is very similar to what we do with livestock. We look for the few genetic variants that become predominant or present where each stress has been frequent over the years. So we look for the few genetic variants that have a spatial distribution that looks like something like this. So where we have a presence of the genetic variant where heat stress has been frequent and an absence of the genetic variant where heat stress has been absent. And as you saw yesterday, we can then construct this genotype environment association model to show how this frequency of the genetic variant responds to the gradient of thermal stress. So keep in mind this image because later on I will show you what a real genotype environment association look like in coral. So this is all theory. Now back to 2016 with Stefan, we had to find out a way to put all these ideas in practice. And this is why we conceived this project, which is called SABLE, which was kind of a pilot project to study coral adaptation using seascape genomics. And we set up this study in New Caledonia. So Stefan was the leader of this project together with Véronique Berthollet-Sellier from the French CNRS and together with Dr. Gaël Lecellier from the Institute of Research for Development of New Caledonia. So together with this team, we conceived this project to see if seascape genomics can actually work to study coral adaptation. So just to give you an idea of where New Caledonia is, so this is Australia, this is the region of the Southwest Pacific. New Caledonia is located just here. It is next is a neighboring reef compared to the Australian Great Barrier Reef. It's important to note that this is a French territory in the middle of the Pacific. It's the second world largest barrier reef in the world. So the first one is the Australian one, and in New Caledonia we have the second one. And the state of the reef of New Caledonia is extremely good. It's an extremely pristine state, the reef, which is quite surprising because they are located just next to Australian Great Barrier Reef, which has been degraded massively over the past decades. So it's a very nice playground to study the adaptation of corals to climate change. So I'm going to show you an overview of the different steps that we took in this study, and then I'm going to discuss a bit more into the details how we performed each of these steps. So we started with this environmental analysis to characterize what environmental variation has been like across the reef of New Caledonia over the past decades. And based on this analysis, we selected some reef exposed to extremely contrasted environmental conditions. We went to the field, we scuba dived, and we collect corals for genetic analysis. At this point, we had a characterization of the reef environment for one side, and a characterization of the genetic variation of the corals that live in this reef environment. So we put the two things together and we ran the genotype environment association study. And then based on the model of association between genes and environment, we tried to predict what will be the distribution of thermally adapted corals across the entire reef system of New Caledonia. And at the same time, we ran some steps of validation just to make sure that these genes that we find be associated with thermal resistance truly confer a thermal resistance in corals. So the first step was to use this environmental analysis to characterize the reef environment. So there are different databases available online that provides access to data about the state of the reef. One of the most used is Copernicus from the European Commission, but we have some equivalent in the United States. So this database gives access to information on different environmental variables. It could be water temperature, water turbidity, solar radiation, or salinity, all around the world over the past decades. And then we can use some geographic information system processing to calculate averages and standard deviation for every single of these environmental variables over the past decades. So just to give you a concrete example of what this can look like, here, for example, for heat stress, here we have New Caledonia, and we have what heat stress has been like in New Caledonia on the 1st January 1985, and we have the same information for the 2nd January 1985. So in short, we have this for every day until today. And then by using some geographic information system, we can find out which of these pixels correspond to coral reef. So every point in this map here corresponds to a coral reef. And the color corresponds to how frequently over this period of more than 30 years, coral reef have been exposed to stressful environmental conditions. So here you can see that the reefs located in the west of New Caledonia have been exposed to recurrent thermal stress, which is not the case, for example, here on the east coast of New Caledonia. So this was a bit the starting point, and this was done in the beginning just for thermal stress. And you have to imagine that we did this for many different environmental variables, such as water pH, salinity, water turbidity, and so on. At the end, we managed to obtain something like this. So here you see the reef of New Caledonia that are classified in different environmental regions. So for example, the east coast of New Caledonia was classified as a region with low thermal stress and low turbidity and low salinity, whereas, for example, in the south, we have a higher thermal stress, a lower salinity. So every environmental region is a region where the environmental conditions are unique compared to the rest of the reef system. And as Elia showed yesterday, we used this analysis because when you manage to understand or characterize the environmental variation, then you can find out what are the locations that would maximize the environmental contrast. And so you will improve or enhance our capability to detect adaptive signals, which is actually what we did. We used this characterization to establish 20 sampling locations all around the reef of New Caledonia, so that we could really go there sampling and enhance to the maximum the statistical power of the downstream analysis. So in the end, we selected these 20 sampling locations. So you see the purple points here on this map around the New Caledonia. And then we went to the field. So we focus on 3 coral species. One is called Acropora millepora, which looks like something like this underwater. And the other 2 species are 2 species that are closely related and they look like something like this when they are underwater. And in total, so we had 20 sampling locations, 3 species. We collected roughly 800 samples of corals, and we travel all the way from Numea to the different locations. So we have to travel a lot around this island, which is actually quite big, as you can see. And in total, the field campaign lasted 3 months. Just to give you a bit a taste of what it looks like in practice, a field campaign in Seascape Genomics. So here you can see the preparation of the field work, so the preparation of the boat with the scuba diving tanks, as well of course, all the sampling material to collect and store the coral samples once on the field, the sheets to record the coordinate of every sample that we collect. Here you can see the team that helped us to collect these corals. So this is the team from the IRD of Numea that helped us to go to the field to navigate across the reef of New Caledonia. And then we usually left the harbor. We travel up to 10 to 20 kilometers from the coast, because in New Caledonia, as you can see, sometimes the reef can be located far away from the coast. We go underwater, we go scuba diving, we look for the species we are interested into. As you can see here, we put them in a tube and then we bring them back to the boat. So we don't sample the entire colony, we just snap a little branch of the coral, as you can see here on this image. And in this amount of tissue, there is sufficient DNA to perform the seascape genomic analysis. So at the end of this field campaign, we collected between 700 to 800 coral samples. We brought them back to the lab in Numea, and we started isolating the DNA of the samples. Well, this is probably all stuff that you know very well, how to perform DNA extraction and genotyping and so on. So just a note, we use the genotyping technique, which is called DART sequencing, which is a technique related or very similar to a RAD sequencing technique. So in the end, we genotyped roughly 400 samples across the 3 different species. And for each of these species, we were able to genotype roughly 10,000 genetic variants, so 10,000 SNPs across the 3 different species. And then we started the genotype environment association analysis. So we started to evaluate which of these 10,000 genetic variants are associated with variation in thermal stress. So I'm going to show you just one example of what one of these significant variants looks like. So a variant that is associated with thermal stress. So you can see it here. So these are, again, the reef of New Caledonia. The color in the background is the frequency of thermal stress around New Caledonia. And you can see here, these little dots, they are a bit modified in this slide, but if we zoom in, this is what they should look like in practice. So basically, we have a sampling location with the corals, the different colonies that we collected from this sampling location. And you can see that here, along the west coast of New Caledonia, where heat stress has been very frequent over the past decades, for this specific genetic variant, we observe a high frequency of the variant on the west coast, but the same variant is very rare or less frequent on the east coast at reefs that have been rarely exposed to thermal stress. So in practice, when we build a true genotype environment association model for this particular genetic variant, we observe something like this. We observe that the frequency of this variant increase in corals that come from reefs that have been exposed to recurrent thermal stress over the years. And in the end of this analysis, we were able to discover between 10 to 20 genetic variants in the different species that follow in each of the species that follow this behavior. So out of thousands of variants, just 10 to 20 showed this association with the gradients of thermal stress. And in the end, it looks like something like this. So for the first species, so Acropora millepora, we have different models of adaptation showing this association between genotype frequency and the frequency of thermal stress. And we had the same thing for the second species, for other genetic variants. And again, for the third species, we had the same thing. So in total, we have roughly 20 genotype environment association models per species. So in total, 60 models. And what we did next was try to calculate an overall average. So a model that averages all these curves, which is what you see here. And this is what we refer to as a model of thermal adaptation for corals from this region. So of course, it's just based on 3 species. So there are many more that should be included in future studies, but it was a bit the proof of concept that mattered for us at that moment. So here you can see what the frequency of all these different genetic variants, how this frequency responds to a variation in thermal stress across New Caledonia. And you can imagine now what the next steps will be, because we know how the frequency of adaptation responds to the frequency of thermal stress. And we also know how the frequency of thermal stress is distributed across the entire reef of New Caledonia. And so by combining these 2 information, we could produce something like that, which is a map of the expected frequency of this genetic variant for the entire reef system of New Caledonia. So the idea behind this map is to estimate a probability of adaptation for the corals of New Caledonia for any single reef. And as you can see here, for example, in the west coast of New Caledonia here, so I flipped a little bit the map, you can see that we have a high probability of adaptation because heat stress has been frequent over the years, and we know that this degree of heat stress is likely to trigger adaptation based on this model. And the opposite situation is on the other coast of New Caledonia. We have a a low probability of adaptation to thermal stress because the frequency of thermal stress is lower. So all of this looks very nice, but there is still a problem, which is something very important that Elia mentioned yesterday, which is that these kind of models, or more in particular, this kind of model are very subject to false discoveries. And sometimes we are not sure that what we discover with these correlation methods are true signal of adaptation or not. And so we ran some runs of validation to find out how much can we believe in the fact that these genotypes are really implicated in the adaptation of coral to climate change. And we have at least 3 good reasons to believe that they actually are. So the first level of validation is that we run an in silico analysis, so an analysis of the genetic annotation of this genetic variant. We looked at what is the genomic position of the variant and which genes are likely to be influenced by this mutation. So what are the genes that are located in proximity of the genetic variants? And the genes that we found often refer to mechanisms such as DNA damage repair, protein folding, or antioxidative mechanism. So these are all molecular functions that are well known to be implicated in how the coral cell responds to thermal stress. So this is a good sign because it is possible or likely that this mutation influence some mechanisms that are key for how the cell perceives thermal stress. Then there is a second level of validation that we run, which is ex situ, which is very new. These results haven't been published yet. But in short, we took some of the corals from the field and we put them in an aquarium and we turned up the heat. And at the same time, we genotyped these corals. And what we wanted to see is how fast do corals bleach in response to the genetic background that they have. So basically, we wanted to check whether the presence of the genetic variants that we discovered through Seascape Genomics conferred some kind of resistance to the experimental exposure to thermal stress. And what we observe is that indeed, corals that carried the genetic variants that we discovered with Seascape Genomics showed reduced bleaching and mortality in these aquarium experiments. So this is a new work that will be published probably in a few months, but that shows the power of Seascape Genomics to uncover variants that are likely to be implicated in adaptation. And then there is a third level of validation for these results, which is the in situ validation. So basically, we wanted to check whether our predictions on the probability of adaptation match the observation from field monitoring surveys. So the idea is really to see whether what we predicted here is what actually happens on the reef, if this reef here hosts truly corals that are more resistant to natural thermal stress. So you have to know that in New Caledonia, there are these NGOs that year after year, they go to the reef and they try to verify what is the state of the reef after different heat waves that come by year. So the idea was to check whether how these different corals respond. So what they do, these NGOs, they measure the abundance of coral and then try to track whether a reef is increasing in the amount of coral or decreasing. And what we observed in this study is that for the coral reef that are located here on the east coast, so the ones that have a low probability of adaptation, every time there is a heat wave of high intensity, we observe that the coral abundance decrease along with the intensity of the thermal stress. But at the same time, when we do this same analysis on other coral reef in yellow, for example, in this map, which are predicted with an intermediate probability of adaptation, we observe that this decline in abundance of coral is much lower. And when we focus on coral reef in green, so the one from the west coast mainly, where the probability of adaptation was very high, we observe that even when there is a very strong heat wave, so when natural thermal stress is very strong, we do not observe a decline of coral. So this is another good sign showing that the prediction that we performed about the probability of adaptation of coral could actually, or at least part of this prediction, could actually be a true biological signal. So I think that this shows a little bit that Seascape Genomics has at least a role to play in how we can find thermally adapted population. Now the next question is how can we use this information concretely to empower coral reef conservation? And one important aspect when we talk about conservation and adaptation is to understand or define what the potential of adaptation truly is. Because based on what I just discussed right now, we might expect that if we have a coral reef that has been exposed to recurrent thermal stress, we should expect that this reef hosts thermally adapted corals. And to the contrary, we should expect that if a reef has rarely been exposed to thermal stress, it should not host or has lower frequency of thermally adapted corals. However, as you probably know, corals, as many marine animals, they reproduce by dispersing larvae in the water column. So coral release the larvae here, and then they are dragged by sea surface currents. And these larvae can persist across hundreds or dozens of kilometers of distance. So through dispersal, coral can settle up to 50 to 100 kilometers from the location from where they were released from. So this is important because we could actually find thermally adapted corals, even at reef, that have never been exposed to stressful conditions. And this is the reason why when we talk about the potential of adaptation of corals, we talk about adaptation, so the probability of adaptation, so the map that I showed you before, but as well about connectivity. And I don't have really the time to develop this into the detail, but the idea is that we can estimate connectivity using the seascape genomics method again, because in seascape genomics, we can estimate first using the environmental characterization, how surface water has moved around New Caledonia for the past decades. So we can take all these satellite images and process them to produce what we call a transition map of sea currents, which is what you see here. So basically, this map shows us, on average, in which direction water moved around New Caledonia over the past decades. So you can see, for example, that here inside the lagoons on the west and on the east coast of New Caledonia, water flows predominantly in a northwestern direction, whereas as we go out of the lagoon, water flows in the opposite direction in the southeast. So based on this transition map, we can estimate the cost of transition between reef. For example, we can see that if we release a coral larvae from reef A to reef B, there is a very high probability of transition because this movement follows the direction of the currents. And on the other hand, if you want to go from the reef A to the reef C, this movement is very unlikely because it will go all the way against the currents. So it is really unlikely that that coral released here will settle on this reef on the east coast of New Caledonia. So what we did is to build these transition matrices and then to see if they matched with the genetic data that we collected in New Caledonia. So basically, if the transition cost between the reef corresponds or match the genetic distances between the reef where we sampled. And we actually saw that this was the case. So we saw that this map really represents how the reef of New Caledonia are interconnected between each other. So we developed this further and we estimated the connectivity patterns all around New Caledonia. So to simplify what these connectivity patterns looks like, they look like something like this. So these are arrows that shows mostly in which direction water flows around New Caledonia. And so now you can see that really in this map, we have two information. We have the information about the probability of adaptation, so the color of the reef, and we also have the information on the connectivity. So we have the two elements that can define the adaptive potential of corals. And now the question is, how can we use, or what are the conservation strategies that could benefit for this information? And there are mainly two conservation strategies used in coral reef research. The first one is very famous and is similar to the one we use in terrestrial area, is the establishment of protected area. So these are areas that cannot protect for climate change because you cannot stop water to becoming warm, but you can protect corals from any kind of local stress related to the human activity. So one of the ideas that we could use this information on the adaptive potential is to move this protected area to protect the source of heat-adapted corals in a reef system, because these are the corals in the end are going to survive probably in the years to come, and we have to make sure that these coral are preserved from any kind of local activity, for example, for fishing activities or human pollution. And a second conservation strategy that is very commonly used is the establishment of coral nurseries. So these are kind of underwater gardens where corals are grown in a protected environment. And the idea of this coral nursery is that corals are grown there so that once they become sufficiently big, they can be transplanted to a reef that has been damaged, for example. And this is a very common strategy. And one of the ideas that we have is that we could start to choose which coral breeds do we grow in these coral nurseries. So instead of growing any type of coral, we start growing corals that are adapted to climate change. And then we could transplant them at location where these corals are absent. So in practice, if we go back to the map that I showed you before, we could see, for example, that here around New Caledonia, here in the southwest, we have a very good location for a marine protected area, because here we have a high probability of adaptation according to our prediction. And we also have, we are located downstream or upstream of an oceanic current. So if we protect the corals here, automatically we protect all the coast, because we protect thermally adapted coral that could, in theory, repopulate all the reef located upstream in the years to come. At the same time, we could also take some corals from this reef that are thermally adapted and transplant them here on the east coast, because we see that here we have a low probability of adaptation. So we could take some corals from here and transplant them here. It is unlikely that they will arrive there following the natural currents. And so by doing so, we could enhance or increase the probability of adaptation or the adaptive potential of this reef located in the eastern part of New Caledonia. So again, this is theory, and this is now piling up somewhere in the scientific literature. And one of the problems that we have now is to find out how can we transpose this information in practical conservation actions. And it is for this reason that at the end of this SABLE project, we created the MANACO network in the Southwest Pacific. So the MANACO network is an international network of coral reef scientists, stakeholders, and policymakers. You can see here the founding members of the MANACO network, and here in this map are the members or where these members come from. And you can see we have a large representativity in the Southwest of the Pacific. So the goal of MANACO is very simply is to facilitate the way we transfer knowledge from what we do in academic research into the practical field of conservation. So help people that every day protect the coral reef in use at least some of the information we can produce using, for example, seascape genomics. And one of the concrete example of the activity that we do is this web tool that we developed with Stefan over the past few years. This is called the Reef Environment Centralized Information System, or RECIF. And RECIF is kind of a Google map of coral reef environments. So it's similar to BioClim, but specific to coral reef. And the idea behind RECIF is to give access to high quality information on the reef environment over the past decades. As you can see, there are different environmental variables related to the water chemistry, all the human activities. And the idea of RECIF is really to give access even to people that is not expert in GIS to use information on the environmental information of the reef. And of course, RECIF is available in complete open access to any coral reef stakeholder around the world. All you need is just an internet connection to log into the website. And then there is also a second type of activities that we do through this MANACO network, which are the consultancy activities. This year in January, for example, we went to the Western Indian Ocean and we tried to set up a seascape genomic experiment working alongside with the locals from these locations. So here you can see when we went to the field and we discussed with local stakeholders, so people or NGOs and local researchers that are involved in coral reef conservation. And from their side, they shared the local knowledge about their corals. And from our side, we shared the knowledge that we have about how genetics can be used to protect coral reef. And at the same time, we organize some training for local researchers so that they can learn how to use R and how to use the software that we developed to run the seascape genomic analysis, and also the laboratory techniques to perform the DNA extraction of the coral, so that when we leave, they should be able to run a seascape genomic experiment all on their own. So this is a bit what I had to say for today. So just a bit of some perspective about what are the next steps of this seascape genomic approach. So what I'm doing right now at Carnegie Science is to try to upscale the seascape genomic approach. So the Stable project was really a first step, really a proof of the concept that seascape genomics can be used for this. But now we have to expand it to different species. We have to expand it to different regions. And so this will be a bit the goal for the years to come. And at the same time, we want to try to validate seascape genomics with additional approaches. And at Carnegie, we have one of the main experts, or the pioneer of the use of CRISPR in corals. And so together with him, we want to develop some assays to make sure that if we turn off a gene that is implicated in coral adaptation, we truly see that the coral cannot adapt anymore. And so this is a bit the work that we are going to perform in the next years. And again, we want to keep on involving local stakeholders in the creation of knowledge on their corals. There is a very nice project going on at TPFL, which takes place in the Red Sea, which is called the Transnational Red Sea Center, where scientists are trying to team up in this really particular area on the geopolitical point of view. And the idea is to bring science, to bring together all these different countries under the banner of coral reef conservation. So thank you very much for your attention. And if you have questions, I'm happy to answer.
Thank you very much, Oliver. So we have time to take 1 or 2 questions from the audience.
So Oliver, I really enjoyed your talk very, very much. Thank you very much. I have a question. Has the algae any role in helping in this resistance to the bleaching, to the heat stress?
Yes, this is a good question. And there are many groups that focus on this because the algae probably plays a role. So it's not just one type of algae. There are at least 4 different species of algae that can live inside a coral. And inside every of these species, there are hundreds of clades. And research has shown that there are some species of this algae that are more resistant. So yes, the algae definitely plays a role. And in truth, this is one of the next steps as well of seascape genomics, is not to consider the coral just as the animal, but the animal plus the algae. The problem is that the algae has a genome that is twice the genome of the coral. So yeah, we have to find out a way to afford all this sequencing then.
Another question, Marco?
Thank you. So technical question about the connectivity part. Can you tell us more on how you related the genetic connectivity to that map of connectivity priorities?
Yes. So I think in this case, we use the RDA. So using RDA, we based RDA not on the outlier, but on the neutral part of the genetic variation. And then we constructed what are called the Morgan eigenvectors, you know probably them. And we built, so the Morgan eigenvector are variables that describe how samples or how location are close to each other in the space. And we calculated these vectors based on sequence. So we used the distance between the sites to calculate the distance based on this Morgan eigenvector. And then we run an RDA and we showed that when we have a variation, a genetic variation, this variation is correlated with a distance between the sites. And this is how we did it.
So with the asymmetric eigenvectors.
Exactly, yes.
Okay, thank you.
Yes.
And we can take a last question here.
Congratulations and thank you for the presentation, really interesting. I have a question about the larvae. Do you think that the larvae that are produced by individuals that are already adapted can already be competent to express the genetic material to address the pressure induced by climate change or temperature warming and so on?
Difficult to know. This is one of the problems of seascape genomics. We cannot really answer to this question. One of the good things is that many aquarium experiments are actually performed on the larvae, and especially the CRISPR experiment are performed on the larvae because we cannot wait a coral to become fully grown to run the experiment. So it's probably something that we can verify. But yes, to be honest, I wouldn't know if there is a paper that showed that adapted corals are already adapted when they are really at a larval stage.
Thank you.
Thank you.
Thank you very much again, Oliver.
Thank you.
So the next speaker is Marco Andrello from the National Research Council Institute for the Study of Anthropic Impacts and Sustainability in the Marine Environment in Rome. And Marco will explain to us how we could take benefit from genetics in the context of spatial conservation planning. So Marco, the floor is yours.
Thank you very much. And thank you, Licia, for inviting me to this very nice workshop. I really like it. So in my talk, I will talk about conservation areas. Conservation areas are one of the most important tools to protect biodiversity. This map shows the current coverage of protected areas worldwide. The current cover is 16% and 8% for terrestrial and marine areas, respectively. This is still far from the objective of the Convention on Biological Diversity, whose target is to protect 30% of land and sea areas by 2030. This means that in the next 7 years, we have to double the coverage of terrestrial protected areas and increase nearly fourfold the coverage of marine protected areas. And how do we choose where to put the next protected areas? This choice can be done using a rigorous method that is called spatial conservation prioritization. And the objective of my talk, of this presentation, is to introduce you to this method and then see what genetics can do to help this method. So I will do a brief introduction to spatial conservation prioritization. Then I will describe several examples on how genetics can be integrated into this framework. These examples are taken from a literature review that we published this year. So we begin with some definitions. Okay, we have this acronym SCP that can be referred either to the systematic conservation planning or to spatial conservation prioritization. So the definition of systematic conservation planning is the science of optimal allocation of resources for conservation actions. And spatial conservation prioritization is this systematic conservation planning to identify new protected areas. So strictly speaking, here we deal with spatial conservation prioritization, but the two expressions sometimes very often are interchanged in the literature. Nobody has ever complained about that. So in this talk, sometimes maybe you can hear me saying systematic conservation planning instead of spatial conservation prioritization, or more simply SCP. But okay, so what is it about? It's about identifying new sites for protection according to 3 main criteria. First criterion is comprehensiveness. So comprehensiveness is ensuring that all biodiversity is included in the selected areas. So all plant and animal species, all ecosystem types, genetic diversity, and so on. The second criterion is adequacy, means ensuring that biodiversity will persist in the long term, and practically this is achieved by ensuring that conservation areas are large enough, they are well connected, and so on. And then the third criterion is efficiency. Efficiency means ensuring that the chosen site for protection will minimize the cost of protection. This is because the resources for protection are often very limited. And how is this done in practice? So imagine you want to create, for example, a network of protected areas to protect all continental Europe. So first of all, what you need to do is to define a grid of planning units. So something like that. This planning unit will be the sites that you can choose to protect or not. The number of planning unit and the size that you choose will depend practically on the computation capacity that you have. Obviously, the more and the finer resolution, the better. Then you need to map your biodiversity on this planning unit. So here for this example with 3 species, this table indicates the presence absence of each species in each planning unit. Let's see if I can use this pointer. there. It's the presence absence of each species in each planning unit. In this example, the conservation features are species, but for conservation features, you can use any other thing that you can map, that you can put on a map. So also habitats, vegetation types, and so on. Then you need to define a target of representation for each species, for each conservation feature. This means, so 20% target here means that we aim to protect at least 20% of the spatial range of that species. So 20% of the planning unit that where the species is present must be included in the final network of protected area. These targets are usually defined according to ecological criteria. So, for example, for a species that is under threat or with a small range, you would choose a higher target. For a species that is very common and least concern, then you can choose a lower target of representation. Then the last piece of information is the cost. The cost of each planning unit, this it can represent directly the monetary cost that you have to pay, the price that you have to pay to purchase that planning unit, or it can also represent an opportunity cost, that is the value of all the economic activities that are lost if you take that planning unit and you create a protected area. So if you create a natural reserve, it's clear you cannot build a factory, you cannot build homes. So the values of all those economic activities are lost. That's the opportunity cost. More simply, if you don't have information or data on this economic aspect, the cost of the planning unit can be set equal to the surface of the planning unit. The larger the planning unit, the more costly. Okay, so the objective then is to find the best selection of planning unit that minimizes the cost of protection. And at the same time, you need to ensure that each species reach its representation target. And we can see how it works with a little game, which is available on the internet. If you go to this web page, there is this little game. Here you have the planning unit with each planning unit as a cost. And in each planning unit, there is the abundance of 3 species, the red, green, and blue species. And the objective is to reach this representation target for each of the 3 species. So you can choose interactively the planning unit that you want to protect. And here you see that the amount of species protected increases, and you need to reach this target. So you would choose planning unit that harbor a lot of species. Then I'm doing now fast. So we have reached the target for the blue species. We need the green and the red. So maybe I choose this one, this one, this one. Then I need a red. Let's see if we can find the red like this. So the red is okay. I need the green, this one. So, okay, I reached the target for all planning unit. The total cost of my choice is above $8,000. So if I ask a computer to do this optimally, the computer will choose those planning units in black, and the total cost of the solution is $1,000. So it's much better than what I did. And okay, I did it very rapidly, but I can assure you, if you sit down and try to do it very well, it's very difficult to find this problem optimally. That's why we need the computer. So it's about this little game. So up. Okay, what we have just done with this little game is to solve this problem, which is called the minimum set problem. The objective is to minimize the total cost of protection, or to minimize the total number of planning units, at the same time ensuring that we reach the target for all the species. Okay, this problem can be written in mathematical terms. Which has 2 advantages. The first advantage is that the mathematical notation is unambiguous, it's clear, so it's very precise. Second advantage is that we can program a computer to solve the problem for us. And this is what it's done. Without going much into the detail in this equation, you can see that we have the components of the data. So here we have the cost, the cost of the planning unit. Here you have the representation for each species, and here you have the target of representation. This is one type of problem. There are various versions of this type of problems. There is another one that is important. It's called the maximal coverage problem. It's a little bit different. Here, the objective is to maximize the number of species meeting the target, and at the same time ensuring that the total cost is below a fixed budget B. This is useful because very often it's difficult to reach the representation target for all the species. So you aim to maximize the number of species that reach the target. And because very often we have a fixed budget for protection. It's the case, for example, when we say, okay, we need to protect 10% of our land. So you have a fixed amount, a fixed budget. Okay, so these problems are solved by the computer. There are 3 software that are used to solve these problems. One is Marxan, and it's the one that we have just seen in the website. The solution was found with this program Marxan that is developed by an Australian team, by Hugh Possingham. Then Prioritizer is a very popular tool that runs in R. It's an R library, so it's convenient if you want to interface it with other type of data, genetic data, for example. And then there is Zonation, is also very common with a graphical user interface. This time it's developed by a Finnish team, by Atti Moilanen in Helsinki. All right, the outcome of this software is the solution of the problem, that is which planning units are selected for protection and which others are not. And this can be represented on a map, like here, where the red areas are the planning units that are selected for protection. Okay, so after this brief introduction to spatial conservation prioritization, let's see how we can use genetics, how genetics can be integrated, and what benefits we can gain from genetics. The first consideration we can do is, in the example that we have seen, we have used species as planning unit, but species are taxonomic units that sometimes are very poorly defined. Sometimes we discover new species, their identification is often uncertain. And then taxonomic differences do not always reflect well the diversity between species. And so, for example, if you want to, let's see, you protect a site with these bears, the American black bear and the Asian black bear. If you protect that site, you will secure a certain amount of evolutionary history. But if you protect the site with the panda, then you will secure a much higher amount of evolutionary history. So this evolutionary history is an important facet of biodiversity. It's something that we want to protect. And so we can use this framework of spatial conservation prioritization. We can adapt it to maximize the amount of evolutionary history rather than the species diversity. Okay, how do we do it? There is a nice example, a nice study that did that on mammals at the level of all the world. The study was done by Dan Rosauer and collaborators. So their aim was to protect all the species of land mammals, so nearly 5,000 species at the global level. So they had something like 12,000 planning units. And they wanted to protect their evolutionary history of the land mammals. So they used the published phylogeny for mammals. Then to build their problem, they used the branches of the phylogenetic trees as conservation features. So not the species, the branches of the phylogenetic tree instead of the species. The advantage of using the branches is, first of all, we do not double count the common branches. So this branch A is common to the 2 species, and if we put it independently in the table, we are not double counting it. So it's better. And then you can set targets according to the target set for the species. So for the terminal branches, it's easy. You can set a target equal to that species. For the inner branches, you can also set a target that can be set equal to the minimum of the terminal branches that are out of that branch. And then we can set weights. So weights are used to give a penalty if you don't reach a target for that conservation features. And here, what we want to do, we want to set higher weights for the branches that are longer, because longer branches are more important. They harbor more amount of evolutionary history. So they did that using the branches, and then they set the planning problem as a maximal coverage problem. They aim to protect 10% of all the world. This is the result. So these are the chosen sites for protection. Here you also have a color. The color represents how often these sites were chosen in different replicas of this exercise, 1,000 replicas. So in red, you have the sites that were chosen most often. These are the irreplaceable sites. Irreplaceable mean that they are critically important for protecting the diversity of mammalian lineages worldwide. They are also widely scattered because it's the phylogenetic diversity of mammals is itself widely scattered by biogeographic forces. And then you have the other colors. So the orange, yellow, and blue, these areas are less irreplaceable. It doesn't mean that they are less important, but just their selection frequency, the lower selection frequency means that there is choice. So I can protect one or the other. And so it represents a sort of freedom of choice of which areas I can protect to have an efficient solution. Okay, they did something interesting. They wanted to see if running the algorithm only with species would find the same solution. So they compare the two, the phylogenetic base with the species-based algorithm, and they found that most selected areas were the same. So the green areas were the same, but there were some areas, those in red, that so important for the conservation of evolutionary history, that were missed by the species-only SCP. So this means that the spatial distribution of species is not a good surrogate for the phylogenetic diversity and for finding priority areas for phylogenetic conservation. Okay, so this study, I think it's a nice example on how we can use this framework, spatial conservation prioritization, to protect conservation features other than species, okay? And this is important also for the conservation of within species diversity, intraspecific genetic diversity. We know intraspecific genetic diversity is important because it underlies morphological diversity, physiological diversity between individuals. It has consequences for population dynamics, for communities, for ecosystem services, network contribution to people, and so on. It's also the raw material for evolution because the adaptive potential of a population is dependent on the amount of intraspecific genetic diversity, and is also declining as a consequence of the loss of range of some species. So if you lose portion of the range, you also lose the local adaptation. So you lose intraspecific genetic diversity. And so the target of the Convention on Biological Diversity is to maintain at least 90% of intraspecific genetic diversity by 2030. And this is not only for domesticated and cultivated species, but also for wild species. And so how to protect that? So in the rest of the presentation, I will show several examples. So now you can use the approach of SCP to integrate intraspecific genetic diversity. Okay, so one approach is to use the same phylogenetic approach with trees, and I will illustrate it with an example on amphibians and reptiles in the Iberian Peninsula. And this study was done by the team of Silvia Carvalho. And so they built interspecific phylogenies for amphibians and reptiles. And then for some of these species, for the ones for which they had the data, they built intraspecific trees, so genealogical trees. So the example here is on the pond turtle, Emys orbicularis. So they built this intraspecific tree, and then they identified conservation units into this tree. For example, here they found 5 conservation units, okay? And they were working on a limited sampling grid. So the black dot here, I don't know if you see them in the map. The little black dots were the sampling sites. So from these sampling sites, with the information on this clustering, then they built a distribution for each lineage. So here the colors that you see is the spatial distribution of the lineage. And you need that because then you need to map the distribution of each lineage in the landscape. And then they built their data set. So as before, we have the branches of the phylogenetic tree as conservation features, and you also have the intraspecific lineages as conservation features. Then they set a target for each of them, and then they set weights for the branches and for the lineages, as I showed before. And then they did 4 scenarios. In the 4 scenarios of conservation planning, in the first, they did the traditional using only species, then another one with species and intra-specific lineages, this one with species and interspecific lineages, and the 4th one combining everything. And then they compared the solution. So now I'm showing you only one comparison between species and intraspecific to see what they obtain. Here is what they obtain, the map on the left on the screen. So the green areas are those selected in common between the 2 scenarios, and it's nearly half of selected areas. So it means that the other half is specific to the scenarios that you use. And in particular, the yellow areas are those that are selected only when you use the intraspecific information. So this is very important. It means if you don't have information on intraspecific diversity, it's very difficult to identify the areas that protect the intraspecific diversity. And then they ask, are these areas, are these important areas for intraspecific diversity? Do they correspond to the location of high local intraspecific diversity? So what is shown here on the map on the right, it's the richness of intraspecific diversity. And the answer is no. The answer is no, because these areas are redundant. So if you protect all these red areas, you would end up with a solution that is not efficient. There are too many. The SCP approach ensures that you can reach the most efficient solution. So you select areas that are complementary in their composition of intraspecific lineages. So this very nice work on amphibians and reptiles show that we can use this phylogenetic approach to protect intraspecific diversity. There are other approaches. So the one that I'm going to show you now is based on genetic metrics. And this is a work to which I had the pleasure to participating. And it was done on the sea cucumber in British Columbia, so the Pacific coast of Canada, by Amanda Xuereb and the University of Toronto. And what Amanda did, so she sampled 24 sites for a total of 717 individuals. Then she used DDRAD sequencing, and she was able to identify nearly 3,700 SNPs. And then she did redundancy analysis that we have seen yesterday to identify which of these SNPs can be considered putatively adaptive. Putatively adaptive to sea bottom temperature. And she found 51 SNPs putatively adaptive, while the other ones were considered neutral. So with these 2 sets of SNPs, neutral and adaptive, we estimated a set of 6 metrics. So we had the heterozygosities, which measure the within site genetic diversity. We calculated these 6 variables at the level of the sampling site. So an heterozygosity represents the within site diversity, then a local FST to represent the between site diversity. And we did that on the neutral set and on the adaptive set. And then we calculated two additional metrics of adaptive variation. The first one is the population adaptive index that was proposed by Aurélie Bonin in 2007. This is the difference in the frequency of the adaptive allele between the focal site and the mean frequency over all the landscape. So the adaptive allele here, we were interested in the association with bottom temperature. So one of the allele is associated positively to temperature and one is associated negatively at this adaptive low C. We consider as adaptive the allele that was associated positively because the consideration was that the sea bottom will warm with climate change, and so having an allele positively associated would be adaptive. So the population adaptive index, and then the last metric was what we called an adaptive score in a paper with Stéphanie Manel, which is nothing better than a polygenic score. So it's the proportion of adaptive alleles per individuals, average over the individuals of a site. So we had these 6 metrics at the level of each site. Then we interpolated them over all the planning grid. So already you can see differences between the 6 metrics. For example, if you compare the within population metrics, the within population metric with the between population metrics. So if you compare neutral and adaptive, you can see different patterns. And then we use these metrics as conservation features in conservation planning. So we build our data set with each metric as a conservation features. And we try to solve this problem as a minimum set problem. And we did that initially with only one metric at a time. And what we obtain are these priority maps. And already you can see that they are very different according to the metric that you choose. And there is one striking difference is between these two, between adaptive heterozygosity and the adaptive score. And the adaptive heterozygosity protects regions in the north, while the adaptive score protects regions in the south. And if you think about it, it's not a surprise because the adaptive heterozygosity is a measure of within population genetic diversity, and it's highest when the allele frequencies are balanced. So adaptive allele and maladaptive allele are present at the same frequency, while the adaptive score measure within population adaptedness. So it's highest when you have a high frequency of an adaptive allele and a very low frequency of the maladaptive allele. So it's the opposite of diversity, actually. So this shows, again, the importance of choosing well the metric depending on your objective. If you want to protect the adaptedness, the fact of being already adapted to a condition, or if you want to protect the adaptive capacity, the potential to adapt. Then we try to run this SCP by combining the metrics. So we used weights to weight one metric against the other, and we run it with couples of metrics, pairs of metrics. And what we obtained was, not surprisingly, a solution that were intermediate between the two metrics. And using two metrics is important because it allows you to consider different aspects of genetic diversity at the same time. For example, here, when we combine adaptive heterozygosity and adaptive scores, so it's here, adaptive heterozygosity and adaptive scores. This allowed us to say something about how important we think it is to being already adapted versus retaining an adaptive capacity. And so the consideration is, if we believe the climate models, if they are accurate and they predict a warming in the future, and if we believe that our identification of adaptive loci and adaptive alleles is accurate, then we can weight more the adaptedness matrix. Okay, we are quite sure that this population need to be adapted to this condition. Okay, the adaptive score. If, on the contrary, there is uncertainty in climate projection, or if we are unsure about the identification of adaptive alleles, or if we don't know their actual effect on fitness, then we can weight more the adaptive capacity, so the heterozygosity. All right. One little problem about this nice approach is that we have to set targets for these metrics, and unfortunately, this is not at all meaningful to set representation target for metrics, because metrics are not like species occurrences that can be summed to reach a target. So one solution to this problem is what I called a split taxon approach. It's an approach that I did not invent, but I just founded the name, split taxon approach. So how does it work? Let's imagine we have the distribution of a species with the values of a genetic matrix. Here it's adaptive heterozygosities. So we start by classifying the levels of adaptive heterozygosity into low, medium, and high. And then what we do is we split the distribution of the species into 3 layers, where we put only the presences, only the population with low heterozygosity or medium heterozygosity or high heterozygosity. So in doing so, we can set targets. And this time the targets are meaningful because it means we aim to protect 50% of the population with low heterozygosity, 30% of those with medium heterozygosity, and so on. And we can also set different targets for this different distribution. Here, for example, if we consider that the low diversity population need more effort, we can set a higher target. Okay. So this approach on metrics show how we can integrate different aspects of genetic diversity, neutral versus adaptive, within population versus between population, or different ways of measuring this aspect. But so ultimately, all these aspects depend on the distribution of the alleles on the landscape and on their frequencies. So another approach is to focus the conservation planning directly on the alleles. And the study that I'm going to show you does exactly this. It's a very nice study by Ivan Patsvinas et al. On 6 species of freshwater fishes in the southwest of France. So they had these 6 species. They use microsatellites, and microsatellites usually have more than 2 alleles per locus. And so so in this case, the planning units were the sample sites, those dots on the map. They did not use a continuous grid, but it doesn't make any difference. They used those planning units, and then they mapped the presence, absence of each allele onto those planning units. And then they formulated the planning exercise as a minimum set problem, that is choosing the smallest number of sites to protect the maximum of genetic diversity. More precisely, they aim to protect at least 1 occurrence of each allele at the minimum cost. And they did that separately for each species. This is what they obtained. This is the same map, colored according whether the site was identified as important for that species. And so first of all, they saw that the different species had different sets of irreplaceable sites, and few sites were in common for more than one species. So the sites that are circled with the dashed lines were important for 2 species, and there was only one site that was important for more than 2 species, this one. And so this means that the conservation solutions are really species specific, unfortunately. And then they ask another interesting question. So they calculated 3 genetic metrics, the allelic richness, the private allelic richness, so the richness for the alleles that are private to each population, and then the index of differentiation of JOST. And these maps show these metrics interpolated on the landscape, so it makes very nice maps. And you can see they have different distribution, these metrics. And then they ask, are these metrics good predictors of the important sites that we have just seen? And here the answer is again no, except for private allelic richness. Private allelic richness was a significant predictor for the important size. And this again suggests that these irreplaceable sites are those harboring high level of endemism. Private alleles are like endemic species, but not necessarily the site showing the greatest genetic diversity, the greatest allelic richnesses. Okay. Okay, so we have seen all these different approaches to integrate intraspecific genetic diversity. Now I will focus on another very important aspect for a network of protected areas, that is connectivity. All right, connectivity is very important because it allows species to move within their home range and being protected. It allows sourcing dynamic. It allows larval dispersal or seed dispersal. It ensures the spread of genes, so gene flow, the spread of adaptive genes. And also it's important for range shift of species as a response to climate change. So connectivity is a very desirable property of a system, of a network of protected area, and it can be integrated in this planning approach. How? Let's see it briefly. I will not go into the details of the equation, but basically the idea is to take the cost equation and to add another piece that represents a fragmentation cost. So it measures the fragmentation of your system of protected areas. And this fragmentation cost is based on this parameter CVIH. CVIH measures the existence of a boundary between two planning units. So here in this example below, planning unit 1 and 2 share one boundary, so the CVIH will be equal to 1, while planning unit 4 and 2 do not share any boundary, so CVIH will be equal to 0. And by using this formulation, we can tell the algorithm to favor selection of planning units that are adjacent, to favor a system that is compacted, more connected. This can be generalized to situation where the connectivity is not dependent on the physical proximity of the planning unit. So, for example, in marine system, connectivity is achieved by larval dispersal over very long distances, several kilometers, and also it can be directional, asymmetric, or symmetric. And so you can take the value of larval dispersal probability and integrate it in this parameter. So basically, if you have an estimate of larval dispersal, you can integrate it into the approach. And this was done in a very nice study on this very beautiful organism. I really love this picture of this Tridacna clam in the coral triangle. And the study was done by Mariah Baker, who's now at the University of, I don't remember, in England, but so on. So they sampled these species over 27 population, 524 individuals, and then they used microsatellites, and then they did 4 analyses. First, they did a structure analysis to identify genetic clusters, and they found 3 genetic clusters, so the blue, the yellow, and the green. Then they calculated allelic richness, so within population diversity. Then local FST, so between population diversity. And then the last one, the important for us, for what I want to show you, they estimated the recent migration rate with this bias approach. And then they set up a planning problem. They had, first of all, they had a baseline scenario that did not use the genetic information. So they identified 9 habitat types. So high coral cover, medium coral cover, deep coral, rock, seagrass and algae and so on. So this, in the baseline scenario, they just had these 9 habitat classes and trying to prioritize a target for each class. Then they did additional scenarios integrating one genetic feature at a time. So for example, scenario A, they integrated the 9 habitat types plus the 3 genetic clusters. And then they define targets of representation for each of these classes. So again, for scenario A, they aim to protect 30% of each habitat class and 30% of each genetic cluster. In this way, they would aim at protecting the genetic diversity. And then scenarios D was the one where they integrated the migration rate. And so they had the 9 habitat classes, and then they integrated the migration rate, pairwise migration rate, through that approach, that CV parameter, as I showed you before. And so they had the first baseline solution showing the important areas for preserving the habitats. And then for each of these scenarios, they had these other alternative solutions. And the one I wanted to show you is on the bottom right for migration rates. That solution ensures that not only the habitats, but also the connectivity is maintained. As you can see, the sites chosen for protection are not physically adjacent, but this reflects the fact that connectivity can be long distance. So, but it's the best solution that conserves the connectivity of this system. All right. Let's try to summarize all these examples that we have seen. So we have talked about this conservation prioritization problem. It can be formulated as a mathematical problem, so with 2 equations. First equation expresses the cost of protection and the cost of fragmentation. The second equation states that we must reach the target of representation for conservation features. Okay, so we need to integrate the genetic information into this equation. And so we start often from sampling sites and we extract genetic information. For example, we measure genetic diversity, or we can estimate dispersal. We can also estimate population size. We have not seen an example on this, but you can estimate effective population size or also census size. And then there is often a step of specialization because you need to transform your information on the sites on information on a continuous grid. After this step, you can integrate this information into the planning problem in various ways. The first as alleles. You can use the alleles as conservation feature, as in the example on freshwater fish that we have seen. Or conservation units is the case when you use the branches and the lineages of your intraspecific phylogeny, or when you use genetic clusters as conservation features, as we have seen in the amphibian and reptile example. Or you can use genetic metrics, as in the sea cucumber example, where you can choose different metrics to measure different aspects of genetic diversity. You can use the split taxa approach to avoid that problem of setting representation target for different metrics. And then something we have not seen, but is, I think, very interesting, if you have estimates of population size per site, you can reformulate this problem not in terms of presence absence, but in terms of abundances, and try to solve to reach the maximum abundance of your species. Okay, then the connectivity and dispersal estimates or gene flow like FST can be integrated to that parameter CVIH, like in the CLAM examples. And then an approach that we have not seen is to integrate the genetic metric as cost. And this, for example, if we think that is more costly to protect sites with a low genetic diversity, we can set a higher cost for them. And then, okay, then there is another approach that I have absolutely no time to talk about it today, but it's very nice, and it's called the environmental diversity approach, and it makes use of continuous measure of genetic variation. So, for example, you can use the axis of your PCA or of your DAPC as a continuous metrics and integrate directly those into the planning approach, because sometimes it's very difficult to identify genetic clusters. Sometimes you don't have a discrete clustering structure. So the environmental diversity approach, and a good reference is the paper by Jeffrey Hanson, is a very, very nice approach to integrate genetic diversity. And so in this review, we have identified only 25 papers published focusing on integrating intraspecific genetic diversity into SCP. So the subject is quite new, so there are a number of open questions. First, what is the added value of all this genetic information? Or put it in another way, what is the risk of disregarding genetic information in spatial conservation planning for biodiversity persistent? Conservation decisions have been made and will be made in the absence of intraspecific genetic data, especially when these data are very costly, demanding to be collected. So in which cases is it worth to spend more time and data to collect, more time and money to collect genetic data? Then second question is, as this type of data are very demanding to be collected, are there proxies, surrogate for intraspecific genetic data? So I'm talking about maybe environmental variables being surrogate information for adaptive genetic variation. There have been a couple of papers focusing on this question. One paper said, yes, it is possible. The other paper said no. Both papers from the same author, so it's interesting. But so it's a question that we don't know yet. We don't know. Then there is that spatialization step. And so this is something you have to do very often, but there are different methods to do that, kriging, interpolation, and so on. Nobody has ever focused on studying the strengths and weaknesses of these methods. Then, very important question, what will happen if we want to protect genetic diversity? How much more land and sea areas, surface areas we need to represent intraspecific genetic diversity? So previous studies have shown that to protect interspecific diversity, so phylogenetic diversity, you would need to expand the protected area system by only 5%. So that was a good finding, but will it be true for intraspecific genetic diversity as well? And then uncertainty, all these estimates and metrics come with an interval of confidence interval or a prediction interval. They are sometimes very uncertain. Your adaptive losses, when you identify adaptive losses, according to the method that you use, you get completely different results. How to integrate this uncertainty into this planning process, and what are the consequences on the solution? And then we often work, so the species that we study with genetics are not a random subset of the species present on Earth. We often focus on species that are nice, that are iconic, that are important for economic activities, that are easy to study. But so what happens if we obtain conservation solution based only on those on that subset of species? What will happen for the other species? So all these.
Sorry, just Marco, I'm sorry, because we also need to conserve time for the coffee break.
Yes.
So we need to come to the
conclusion. So if all these questions, if you find them interesting, this is my last slide. We have a special issue in biological conservation coming up. I will be co-editing this special issue with my colleague, Cassidy Daloya. So if you find this question interesting, you're very welcome to submit a manuscript until the end of February. If you didn't find it interesting, sorry, I hope it was not too boring. Thank you for the attention.
Thank you very much, Marco. So I suggest that we take one question from the audience now, and then the rest can, the other questions can be discussed during the coffee break. So is there any question for Marco? If not, I've won. Oh, sorry, Oliver, go.
So thank you, Marco. Super interesting. I had a question about the spatialization. So I guess most of the time you do it by plain interpolation.
Yes.
Do you know if there is if it would be pertinent, for example, to model each of these different variables, for example, heterozygosity, modeling on many environmental variables and use them as a proxy of for is that this has been done, or is it something that could work?
I don't remember if it's been done, but you can try. And what you can do, there is this package, the fill-in package, I think, by Tarosso et al. And you can try to model these genetic surfaces by using a variogram. And I think you can model them as a function of space. And also if you can try inserting environmental variables, then you compare how well your model performs. And then, yeah, you can try.
Thank you.
Thank you, Oliver. Yeah, I just take 1 minute for this question. That okay? The theoretical background is perfectly mastered. Then how do you move from the conservation planning you propose here to concrete conservation actions, to money allocations to the specific units you identified?
So conservation planning is an approach that dates back since the beginning, the end of the 90s, and it was developed for species, and they also developed a framework for translating all these studies into practical conservation action. So if you read the paper by Margulis and Pressey 2000, they say this aspect is only a step in a large process where you need to identify your species and the data that you need, your conservation problems, and then negotiate with the people. Mm-hmm. So you can go to the table, say this is the optimal solution for the features that I'm interested in. What do we do? And so there are also planning tools that are designed to be interactive. You go to the table, you say, okay, if we do that, and negotiation
with local authorities and
local authorities, stakeholders. Yes.
Mm-hmm. Yeah. Excellent. Thank you again.
So hello, I think we can start with the second session of the morning. This session is dedicated to the LIFE projects, and we'll start with the first presentation by Licia. Licia will explain us how to combine genetic and ecological modeling to improve the conservation of house row of the, let's say, the target species of the project. Thank you, Licia.
Okay, thanks, Elia, for introducing me and the work I will describe this morning. Okay, and please inform me in case when I have 5 minutes left, please let me know in case, okay. Okay. So, as you know, this event has been made possible thanks to the support of the LIFE EU framework initiatives. And among the projects funded by the LIFE framework, there is also LIFE Globe, which deals with species, which is Austapotamobius pallipes, which is the white-clawed crayfish, so a freshwater crayfish species, which is present also in Italy. So through this presentation, I will describe both the project itself, how it is structured and what are its goals, and what part of what we have done so far to improve the conservation status of this species. So Austapotamobius pallipes as a taxon is part of a species complex, which also includes the Austapotamobius italicus taxon, which is present in the Italian peninsula. The LIFE Globe project has as a general aim that of improving, as said, the conservation status of this taxon across the northwestern Apennines within Italy. And the actions through which we are aiming to reach this goal are different in terms of time extension and also type of action. In particular, we are dealing with environmental management on a side, ex situ breeding on the other side, to perform restocking actions aided by molecular knowledge. So the LifeGlow project includes 10 partners, universities, research institutions, national parks, communes, and has a duration of 5 years. It started 3 years ago and is going to finish in 2024. So thanks to the funds we received within LifeGlow, we are targeting the Northwestern Apennines, which basically is a part of Italian country which spreads across the Emilia-Romagna and Liguria regions and the provinces of Piacenza, Parma, Reggio Emilia, Genova, and Savona. Overall, we are targeting 28 Natura 2000 sites of community importance. Just to briefly summarize the many and diverse actions we are carrying out, there are 5 preparatory actions which are devoted to the training of personnel and operators, the population survey of the species in the sites of community importance, the molecular characterization, of which the University of Piacenza is in charge, and also the habitat modeling, which has been carried out in a collaboration between Università di Piacenza and University of Pavia. We have also conservation actions, which regard the containment of the invasive crayfish species, the prevention of disease spread, the ex situ breeding of Australopithecus pallipes to perform the reintroduction of a number of populations in sites suitable from the environmental perspective. We have 4 ex situ breeding centers located where you can see on the map. And here the goal is to become able, along the duration of the project, to reintroduce 10 white-clawed crayfish populations in suitable habitats. Here you can have a look at one of our facilities. This is the breeding facility located in Corniglio, which is in the Apennines in the province of Parma. And as you can see, there is a building, because these breeding sites have been located where buildings were already existing. So we have either taken advantage of pre-existing structures or also performed partial restoration of these structures thanks to the funds of the project. And also we have the pools on the outside of the buildings, as you can see in the picture on the right. Okay, we also have monitoring actions, both for the improvement of the populations and also of the impacts of the project on the socioeconomy of the territory, and also dissemination actions, of which, of course, WIGGIS Workshop is part. Okay, so going back to the actual issue this species is experiencing, the taxon over the Italian country has experienced during the last 10 years a really severe contraction, both in terms of number of individuals and number of locations where the species is present. We are talking about 74% reduction. So in order to be able to effectively perform any conservation action, first we need to answer a few basic questions like those you see listed here. So first of all, we need to understand if the current population of the species are isolated reproductive units or not, and which populations may represent the best sources for the selection of the breeders to be taken to the ex situ facilities for ex situ reproduction. And also afterwards, where should we reintroduce the species so that the chances, that the reintroduction becomes effective are highest. And to this end, we have actions devoted to producing and analyzing data to make these things possible. So coming to the current results of the molecular characterizations, during 2 years of the project, 2020-2021, the sampling has collected almost 1,000 samples from 42 populations, which have been characterized at the level of the nuclear genome through genotyping by sequencing technique, which has many advantages, including that of not requiring any a priori knowledge on the structure of the genome of the species, because there is no reference genome available for Staphylococci pallidus, at least so far. The data have been produced in outsourcing over more than 500 samples, representing 37 populations. And after the typing, the raw dataset included more than 130,000 SNP markers. But after the filtering, the final dataset we are working with includes 467 individuals and more than 15,000 markers. We also sequenced part of the mitochondrial DNA, aiming at the cytochrome oxidase 1 gene as a target, and we have characterized at this level almost all the individuals we had. Okay, regarding the statistical analysis performed on the molecular data, we have basically calculated a set of standard statistics typical of the two kind of molecular markers, nuclear and mitochondrial, by calculating for the genotyping by sequencing data the heterozygosities, performing a PCA, carrying out population structure analysis, and also other analysis to understand how diversity is structured, and also trying to identify possible gene flow events in order to understand how isolated or not isolated our populations are. For the mitochondrial DNA, we calculated the number of haplotypes, nucleotide diversity, and then built a median joining network again to understand how variation is distributed on the territory and among the populations. Coming to the results, the calculation of the heterozygosities in general as a trend highlighted the occurrence of low values, and observed heterozygosity was on average higher than the expected one. The principal component analysis plot, as you see here, shows on the first axis, accounting for more than 50% of the total variation, a very clear distinction between Austrapotamobius pallipes on the right and Austrapotamobius italicus on the left, which are the two taxa known to live in Liguria, so in the western part of the distribution area, or the project area, Austrapotamobius pallipes, and in Emilia-Romagna, Austrapotamobius italicus. So we have a very clear correspondence between the phylogeny and also the molecular variation. When the third principal component is taken into account, two populations from Emilia located in the easternmost side of the project area, they separate from the rest of the Emilian populations. You see them on the top left corner. So here the question we will try to answer in the next month is that is if we are dealing with a different subspecies or else. The population structure analysis performed with admixture software shows or confirms the clear differentiation between Austrapotamobius pallipes and italicus, starting from the very first runs of analysis here. But as we increase the number of genomic components or pseudopopulations, okay, hypothesized by the analysis, we can see that the variation becomes partitioned in a fashion that is geographically explainable. As you see here, when we plotted the average behavior of the population as a pie chart on the population provenance on the map. Okay, this is the reconstruction for K5, so assuming 5 gene pools, or how we prefer to call them, hypothetical populations, and here is the resolution at K9. So again, as we improve the resolution of the analysis, anyway, the geographical structuring of diversity is confirmed. Also, the neighbor network basically confirms the same occurrence. And the analysis of gene flow and migration events allow it to, again, confirm once more the particular behavior of the two eastern Emilian populations, and also to highlight the presence of a number of gene flow events among different populations that you can see plotted as double-headed arrows here on the map for your convenience. Okay, so basically these gene flow events are consistent with the structuring of diversity. In fact, we find the occurrence of the same genomic components shared by the populations between which the gene flow event has been highlighted. Coming to the mitochondrial DNA data, the statistics we calculated show overall the low values of within population diversity, with the occurrence of many private haplotypes occurring in a single population, that is to say, and also many populations, those highlighted in red, which are characterized by a single mitochondrial variant. So there is no variation at all. There is just only a single variant, even though the number of individuals analyzed is not low. This is the network reconstructed from starting from the same data set. It shows the presence of more than 60 different haplotypes, which are mainly population specific. Here the colors represent different populations of provenance. There is one major haplotype which is shared by a high number of populations, the core haplotype of the bottom left part of the network. And there are also a few more variants which are shared by more than one populations which are located over short geographical distances, so they are neighbors. Okay, regarding the two eastern Emilian populations, you can see that their behavior is a bit different from that shown by the genomic data, nuclear genomic data, in that they are located there, the haplotypes they possess are located in quite different parts of the network, okay? So we have the haplotypes from one populations, which are close to the, let's say, the core haplotype, while the second one is located, we may say, a little closer to the Australopithecus pallipes, okay, variation shown on the top right corner. This is the same network where we have adopted the same color coding as in the population structure analysis to show that anyway, besides these two eastern populations, the remaining ones also show a geographical structuring of variation also with respect to the mitochondrial DNA. But also habitat modeling has been performed in order, as said, to guide the selection of the reintroduction areas in the sites of community importance, which you should have seen represented in this map, but unfortunately, I just realized the resolution is not good. I'm sorry. Okay, to perform the habitat modeling, 4 different types of geospatial data have been collected. Land cover, land use data, data on climate, that is to say the 19 bioclimatic variables from the World Clim database, which have been introduced yesterday, and also data describing elevation and the derived topography and hydrography, which basically derive from the geo dataset made available by 5 neighboring Italian regions that surround the project area that you see in the map. Also, information on the spatial occurrence of the species has been incorporated in the model, and this information comes from the Australopithecus pallipes population survey carried out during the project by the colleagues of the University of Pavia and other partners. But also historical information has been incorporated, making use of what was available in the database of the University of Pavia and also records from the region Liguria, covering a time range of around 10 years from 2011 and 2021. As a reference spatial unit for habitat modeling, the single reach was considered as the basis, that is to say the river or stream segment with no lateral tributaries. The ecological niche models were produced using a presence-only modeling method with Maxent software and a related R package. Since the data set was quite complex in terms of number of covariates present, a PCA approach was used to reduce the dimensionality of the data set so that in the end 12 variables summarizing the environmental features were taken to the following steps. What is shown by the model we obtain? The results basically are in line with the ecological knowledge about the species, since the model predicts that the species will most probably occur in small streams in mountain areas and in an intermediate altitudinal range. In terms of variables, as the best predictors, we found variables related to precipitation, temperature, and altitude, while variables describing the habitat had a marginal effect, so they didn't contribute that much, or at least they contributed less to the model overall. So among the factors that disfavor the presence of the species, we derive the following evidence. So the presence is not favored at both the two extreme, we may say, of the range of the temperature values in the study area. The species is not present in the flatland and also at the highest latitudes. A positive effect is had by the occurrence of water reservoirs despite their origin, be it natural or artificial, and also while the occurrence of human settlement plays against the occurrence of Austapotomobius pallipes. The model overall has a good performance, so it is its average area under the curve has a value of 0.777, which is satisfying. And the values of sensitivity and specificity indicated that the model has a good capacity of predicting or classifying correctly absence localities, 84%, while as an intermediate moderate performance when predicting presence localities. The projection of the model over the whole data set of the river segments we had available led to the habitat suitability map you see represented here, in which the predicted absence and presence are highlighted with different color, white and blue respectively. Okay, to start concluding, to start wrapping up with respect to the molecular data, we observed a clear differentiation both at the nuclear and mitochondrial level between Austapotomobius pallipes in the west and Austapotomobius italicus in the central and eastern part of the project area. Overall, we scored a remarkable level of diversity, but within populations, the values of variation were definitely low. Diversity is clearly geographically structured, and as anticipated before, now we have to go more in depth in understanding the behavior of the two eastern populations. Because, of course, understanding if we are dealing with the different subspecies there is important to correctly plan also the reintroduction activities in the proper part of the country. If we consider the phylogeographical patterns that emerge from the data analysis, both type of markers, what's happening? Sorry. Okay. Both type of markers, their behavior points at the impact of paleoclimatic events which have affected the Mediterranean area during the Pleistocene. So we still see the consequences of past population subdivisions today. Regarding the habitat modeling, so the model devised predicts that the occurrence of the species will be mostly concentrated, as said, in small streams and on the mountains where the altitude is not neither too high nor too low. And there is a positive effect when water reservoirs are present, while human settlement, as said, they have a negative impact on the occurrence of Austapotomobius pallipes. This knowledge, of course, was particularly important to take into account if you consider that very, very recently our colleagues have released the newborn, the small crayfish you see in the picture, which come from the first year of ex situ reproduction. So more than 1,500 newborns have been released during the last weeks. And here, I hope it will work. I just want to conclude by showing you a small video. It's a couple of minutes. Is it okay? Do we have time? Okay. Okay. So here you see the ponds outside the breeding facilities, and here are people starting to collect all the elements present in the pool in order to locate and retrieve the small crayfish. And yeah, it's a time-consuming activity which needs to be carried out with a lot of patience and paying a lot of attention because the animals can be very small sometimes. Okay. And then cleaning is needed. Okay, so we can move a bit forward, maybe. Okay, so you see that some hiding sites have been provided in terms of chunks of tubes that were left in the ponds. Then the animals are measured. So there is a record of all the little ones before they are released. And here, finally, the new home. So this is one of the streams identified as suitable for the release of the small crayfish based on the distribution of genetic variation and also the habitat modeling that has been performed. So yeah, they first do not look that comfortable in the new environment, but they have to adapt. Okay, so as a very last slide, the next step to add some perspective to what we are doing, we are going to characterize with GBS more animals and also with mitochondrial DNA. If we have the resources, we would like also to characterize the genome of the species, because if we can do that, we can exploit more fully the molecular information that we are producing. And also we are definitely interested in applying some landscape genomics approaches also to this data set, since it's the number of markers is probably high and individuals is probably high enough to allow this. And the second round of ex situ breeding is currently ongoing. In fact, we are going to collect breeding individuals from 8 to 10 newly selected source populations, so that next year we hope to be able to release a high number of small crayfish across the target area. Okay, thanks to all the partners, to you for your attention, and I'm happy to take questions, of course.
Thank you very much, Lisa, for your very interesting presentation. So any question from the audience? Yeah, there is one question, I think, there. Marco.
Thank you. So it's really a nice project combining different approaches. And so in your next steps, are you going to look at the consequences, potential consequences of climate change? Because if you have the environmental niche model, and if you found that precipitation and temperature are important, you have everything to study that. And there are also, I've seen approaches where you can do the niche model for each of your genetic cluster, and you would have all the data to explore these nice questions. So I was wondering if.
Yeah, this would be definitely interesting aspects to take into account in the future, because so far, basically at this stage, what we are aiming to do is to understand what is out there And afterwards, after we gain this knowledge, also putting the molecular and environmental information we have in a time perspective, okay, with respect to what is expected to happen in terms of climate change in the future, will also be definitely interesting to further define the goals and how and where to reintroduce which populations, if we are able to find also a relationship between the occurrence of specific variants and certain environmental features of the different provenances, population provenances. Julia?
Don't want to monopolize the attention, but I think when you do the reintroduction, you pay attention to which genetic cluster,
yes,
and to introduce, of course. But but that genetic structure, from what I saw, the phylogeography, is I think it's the outcome of maybe a recolonization process. No, but my question is, do you think there is a risk of outbreeding depression if you put the individuals of one cluster in the area of another cluster? Do you think there is a real risk of outbreeding depression when you mix the cluster, or these clusters are just there because history is like that? There is not really an adaptation.
Okay, so well, actually, so far, predicting what can happen in terms of outbreeding depression is difficult. Also considering that it's difficult to relate the variation, the information on the genetic variation we have with the functional aspects of the variation itself. But according to also the previous knowledge on the species, the geographical pattern, okay, of the distribution of variation is likely to mirror what happened in the past, so the paleoclimate of the area during the Pleistocene, because probably what we have two different levels overlapping. One is the phylogeography describing paleoclimatic events, and the other one is the effects of the shrinkage of the distribution area of the species, because for a number of reasons which I hadn't time to list, but problems related to the expansion of crayfish invasive species and also the diseases that were brought in by these alien species, the populations of Austapotomobius pallipes now cover a way smaller area with respect to the past. Okay, so we have the two levels that are overlapping. What was the distribution of variation before, and then what is left after the shrinkage of the populations. So what we are seeing now is probably the combined effect of these two aspects.
Any other question? Yeah, there is one there. I think we have time for one more question.
Okay.
Congratulations. It's very, very good work. As you know, and as you mentioned, we did a similar work 10 years ago, and we found a clear separation between North and South, the border between Emilia-Romagna and Tuscany. Do you have any new data about this, or?
No, not yet, actually, because as part of the next step, we are planning also to extend the genotyping by sequence and mitochondrial characterization also to samples coming from other parts of Italy by widening the area both northwards and southwards, if possible. And also we are in touch with some colleagues working in Tuscany, which have recently received the funding to study the species, and we are going if everything goes well, to exchange samples so that also the mapping of genetic variation will be more extended. Okay.
So I think we are perfectly on time for
Thanks.
passing to the next presentation. Thank you very much, Licia, for your contribution. So next presentation will be held by Claudio Ferrari from the University of Parma. And Claudio will explain how the freshwater fish clean can escape from threats and pressures. Thank you, Claudio. The floor is yours.
Okay, thank you very much for these days, and it's very a pleasure for me to be here, and especially to see plenty of presentation, very interesting. My presentation is a part of the work that I've been done in my PhD thesis, and it's a part of a small part of the Life Barbis project. And I would like to say to title this presentation "Freshwater Fish Climb," because even freshwater fish seems they can climb in terms of they have a kind of ability to escape from environmental condition, and even also for climate change. And how can they escape from threats and pressures? Okay, let's start from a little brief of history of the pressure and threats of freshwater fish species. Okay, in terms of biodiversity in Europe, we know that the most dangerous vertebrates in all vertebrates, the most threats and dangerous species are freshwater fish. Almost the 40% of freshwater fish are endangered species or threatened species, or even who have no data species. And but in our project, we focus on especially native species regarding barber species in Italy, as in particular in the north part of Italy. In the north part of Italy, we have 4 native species. It's a broke barbel, it's called even also canine barbel, common barbel, Balkanic barbel, and Tiberial barbel. I know there are a lot of barbels named, but don't worry. And these fish are very interesting, and it's very didactical. Why is so didactical, this fish? It's so didactical because freshwater fish cannot escape from the climate change, like, for example, the birds or mammals. They live in a very small habitat, in a very small environment, and they cannot escape so much like in terms of strategy as like mammals or birds. So what we know, this is the Barbes is very complex taxon. It's called even also the is like a Latino soap opera. If you want to describe the history of Barbel species, why Latino soap opera? That's we can explain. First of all, the one of the first. Sorry. I cannot. One of the first papers regarding the genetics and population genetics of these fish was from 2013 from Meraner and colleagues, and they describe in a very huge data set the population structure of barbel in the north part, especially of Italy. And they found just 2 species that lives almost together. Okay, one is an alloctonous, an invasive species, is Barbus barbus, and the other is the native one, is the Barbus plebeus. This is represented in this structure analysis in green. And they find out that there is a very a very recent invasion from the east part of Europe of the alien and alloctonous species. And they start to invade the major river that we have. Another papers from Buonerba and colleagues, they from Buonerba and colleagues, they found almost the same results, but in other region, in other sectors, and they found out even other species that lives in this area, for example, Barbus tiberinus and Barbus balcanicus. They are native one, and they described it with the neutral gene, nuclear gene. So the conservation threats we already know about, and they already mentioned in these days, the other colleagues and the other speaker, the loss of habitat, the framework of the rivers, the restocking without any genetic knowledge, and the climate change effect and so on. This is one of the conservation threats and pressure that we have, and we are in front of when we talk about the freshwater fish, especially the restocking without any genetic knowledge was one of the major threats that I want to describe and talk today with you. Because it's a very usually the genetics in terms of management of natural stock, in particular, for example, for freshwater fish, are not so, ancient, as not so traditional, and in most of the case they did it in a very in the past in a very I don't know how can I say it, not properly correct. Even as you know, the sustainability development goals, they have three different goals in the agenda of 2020. That's mentioned the freshwater fish. So they are very important, but they are very important even for this year, because this is the recent pictures of Po rivers, the major rivers that we have, that is almost completely dry. Nothing is happened and was worse than this year, and it seems that year per year it's gonna be worse, the situation in terms of dry season and the warm periods of freshwater for the Po River and the other rivers. So that's why there was we are planning to protect those fish inside a European project. It's called the Barbis project, and the main goal of this project is to create a new population and reinforcing the existing population of this fish, identify threats and local pressure of freshwater ecosystem, because here we are in the most productivity valley of Italy in terms of agricultural systems, and this require a lot of water and a lot of, for example, effort to from the environment. So this reflects even a lot of damage regarding the local stocks and even also the for endangered species, and to try to eradicate and control the spread of invasive alien species. And of course, as a life project, a natural life project, puts guideline for the conservation and sustainability of management species. So in particular, my aims inside my PhD project was to detect, if any, a significant population genetic structure between native species, determining the introgression and the hybridization level between native species and other barbers, and explaining the phylogenetic relationships among population in different hydrogeographic districts. and of course define whatever ecological and environmental factor correlated to genetic factors. So let's talk about the first aspect of molecular taxonomy and distribution of barbed species inside 15 Natura 2000 sites. This is almost the local, very rudimental maps, but it was rudimental maps from Kotelat and Freyo for 2007. And they describe the distribution of these two species, the native species in Italy of barbets, no? And as you can see, of Barbus caninus, they said, okay, Barbus caninus is inside this area where we can find Barbus plebeus and even Tiberinus in the part of Tuscany. Is it real or not? Okay, the first check is was to sampling and collect samples from 27 sampling sites inside these protected areas in the red dots. And the interesting part that we found out that in only one single protected area there was the Barbus caninus. And so if you remember, this map is very, the original areas of Barbus caninus was from the north part of Italy until the middle of Tuscany, but it was not like that in our collected sites. For example, Barbus caninus, yeah, exactly. In this case, Barbus caninus is only one site that was found out. And this is a very, not preliminary result, but the result of Cytochrome B of almost 300 samples. And we found out that the major species that we found out was Barbus plebeus, and the other Barbus caninus and Barbus barbus. Barbus barbus is the Alloctonus and invasive species, as Meraner mentioned in his papers in 2013. They describe a kind of introgression even in the Apennines of rivers. So the first result of this part is we characterize the different the population structure of this of this species in the four rivers for Barbus barbus. In only one site, protected area, there is no evidence of Barbus. There is one evidence of Barbus autochthonous species, but results even show that the alien and invasive species can across the artificial barriers. This is very interesting because sometimes barriers are not capable to stop the alloctonous and invasive species. That's we found out for barb species, especially if these fish are very they are able to swim very well in like salmons or trouts for Italy. They can cross easily the barriers. So, but as you can you can say, and you already know as an ecologist, we know that all of this aspect is very important, but is a system with a systematic point of view. So we cannot see just the genetics aspect, but even also the ecological index, the physical and chemical factors are very important for to choose the habitat suitability, as you mentioned, as the colleagues mentioned before in the presentation, and this aspect can match each other. So that's why we try to collect temperature, pH, conducibility, oxidant demand, and so on. And we found out a very huge amount of difference from the valley to the mountains. So this is quite clear because say, okay, for sure, in the mountains there are less polluted water. Usually it should be less polluted water than the southern part of the river and the valley part of the branch and the rivers. And this is what's happening. Is a very rudimental maps, I know, but in the colors represent the quality of one of the ecological index that we collect and we analyze. For example, ISECI is a very important index for the freshwater fish community. Is the freshwater fish community index, and the important aspect is that the 58% of the sites display a good quality of class. But the 32% is a moderated quality, the 3 poor quality, and the 7 bad quality. But sometimes the index, usually the inside papers are very badly described, and the reviewer, they don't look very well. This why? Because most of the index, they don't speak each other. For example, we can have a very good quality, for example, or a kind of worse quality here, but for the functional fluvial index are quite good. Why this? Because they calculate the different aspects. So that means, for example, for the freshwater fish community, I can have a very good quality of freshwater fish community, but if in that moment I collected the water and one farmers, they throw polluted, for example, they did polluted actions. So for example, to release, for example, trash from his factories, I can collect this water, and this reflects a very a picture of this moment. But they don't reflect in the, for example, freshwater fish community still good in that moment. So that's why usually the ecological index, they don't match each other very well. But that we collect all this information and we try to put on PC analysis, and we found out that just only the alien European barbel species are significantly positive. They have a significant relation with the oxygen demand. And the others, they don't have, it seems they don't have a very strong relation with these factors. But so this first conclusion that I can say that usually the preference of the canine and common barbels, so the native ones, prefer the highest EFF, the fluvial functional index value, and they it's like a proxy of well-developed and structural repair contains and higher altitude value, this one. For the other, for example, for the other techniques and the other characters, we have a higher association level with TSS and the oxygen demands in terms of alien species. But this is just a very brief results of what we found out in this area. Let's talk about more my part, the molecular parts. We select 10 macrosatellite loci for all of these populations, while loci not, for example, the genomic approach, because the barbels have a very huge, a kind of huge problems. So they are tetraploids. okay, is not a problem because they are their nature, they are tetraploids. And this reflects a very interesting history because barbels belongs to the Cyprinids group, the Cyprinids family, and is the worldwide spread family in the world. And barbels that are, for example, in African continent, are tetraploid, are also diploid and hexaploid and decaploids. So they are very, very, very complex taxon. So it's a problem to analyze this one in terms of, for example, structure analysis and other analysis, because just a few software are involved in polyploid in this polyploid aspect. When for example, I asked even in the conference which kind of software I can use to analyze that ones. Sometimes they split the question because the answer, because it's a very big trick. So, but we collect this data and we try to figure out the results with this is, for example, an FCR analysis or correspondent analysis factor. Sorry, of this one I cannot remove this. I don't know why, but anyway, as you can see in this even in FCR, but this is a not multidimensional scale. We have three different clusters that represent the color, which represent all the population that we had. And the interesting part is this one. So the individuals that are in the middle of these three different clusters that present, this is Barbus plebeus and Barbus caninus. Sorry, of this one I want to add, I cannot, okay, now it's better. But we try again with the structure analysis. Structure analysis, it's each of these bars represent an individual, as you know, and the color represent different clusters of this population. We found out again three different clusters, the measure response they give with Evano and colleagues with the Evano's softwares. And we match even with the mitochondrial DNA analysis. Why this one? Because we found out, for example, in this case, that mitochondrial DNA analysis say that this individual belongs to the cluster of Barbus caninus, but the nuclear DNA analysis is different. So we found out 7 individuals like this, and this reflects a very interesting phenomena. It's called the dilution effect of hybridization. What I wanna explain briefly this interesting effect. So if you have 2 different animals, one male and one female, okay, with a different completely genome, they can match. If they can, for example, go they do a reproduction event, and this is the result. So I have a mitochondrial DNA that say I belongs to this one, because mitochondrial DNA they can heritage from the mother, but the nuclear DNA not. The nuclear DNA is half from the mother and half belongs to the father. But if I can go ahead in this way, so I have a female of a hybrid female that can do a reproductive event with male female and go ahead. So this is a very interesting event. It's difficult to identify even if you have a very big data set, because you must be very lucky in the sampling side to catch an individual with a lot of a level of dilution effect. But the hybridization, it seems, is not like blue or green. So it's not like two different completely species. So this is a half of from this species and a half of this. But it's like to ask a person or a people in front of you what kind of color you can see here or here. So you cannot identify very well. So that is another part more regarding the altitude. That's why they can escape from the warmest part of the rivers. So we took the structure analysis frequencies, and we did this analysis against the elevation effect. That we found out even the frequencies that we collect here, we split in the 3 different clusters that we found out. So we found out that there is even a dilution effect, a kind of dilution effect of the nuclear genome, different between the 3 different species. For example, for Barbus barbus, the species that usually is more in the southern part of the river, it's more, in effect, it's more linked to this part of the branch of the rivers, and not in the top of the rivers, except this kind of interesting exception. The Barbus caninus reflect the contrary. So is in the upper, sorry, is this one, the Barbus caninus, is not this one, is the smallest species for bulbes, and it's called broke bulb because it's more in the in the top of the mountains he lives. So we found in their genome more linked to the highest altitude. And this is one of the, I think, the interesting part, the Barbus plebeus. Barbus plebeus can live along the entire rivers, and it seems they have no interest to live more in this Mediterranean part or in the upper part of the river. So just to remind, this must be the situation of the 2 native populations. So in a very good point, in the hypothetical wonderful world, we have Barbus plebeus that lives in the Mediterranean stretch of the rivers, and Barbus caninus lives alone by himself in the northern part of the river. But if we increase the temperature, the Barbus plebeus must move across the entire freshwater fish course and move from the southern to the north. And this is the hybrid zone, the creation of the hybrid zone. But we have another issue. So the Barbus is the alien species. So alien species can move in terms of the highest temperature and the warmest water in the northern part. As you can imagine, for example, if they move ahead in the upper part, we can have the local extinctions. So that's why, if you remember the first paper of Cotteland and Frey of distribution of these fish, reflects these local extinctions. So there are plenty of local extinction in their native areas, due probably for the warmest temperature. So this is a paper. I'm sorry for
Sorry,
this
Claudio.
one.
I think we are going out of the time.
Okay, sorry. I'm finished. Okay.
Yeah, okay. Perfect.
This is another paper that explain better what I said, and it's almost the same. So if, for example, you improve and you increase the temperature, the species A move from the bottom of the mountain to the upper part of the mountain, and the areas and the habitat of species B, they must be they become very restricted. This is the last part of my the analysis. We try to see if in the lateral branch of the major river reflect a different, for example, condition of this species. For example, with the stellar and manuals, I put an order of the rivers, and I want to see if the stream order against to elevation it could be a very interesting match between, for example, the in terms of the nuclear genome results, and that we found out that the elevation and the stream order is very important, especially for the native ones. It seems that the native ones are more linked to the second branch of the branch of the rivers, and the contrary, the second branch of the river is very important for the conservation of the native and pure population. So this is just an explanation of this kind of effect. So Barbus plebeus seems to take all the, for example, it's like a vehicle, a kind of carry to the Barbus plebeus can carry the Barbus genomes, spread the genomes through the hybridization, through the altitude levels. So I can go ahead. Okay, this is for example what we can we did with inside the Barbels project. So we catch the fish, we collect the fish inside the hatcheries. We did the analysis, the genetic analysis to select the spawners, and then we release the spawners through the rivers. So thank you very much for this. I'm so sorry for the time. I'm already finished, and this is what why it's very important the management, the active management against the passive management. So they can reflect the importance. The natural stocks is very important to do the for the native stock. It's very important to do the active management because they can select the habitat manipulation better, and with the good and properly good translocation with the genetic knowledge can restock a native population. Thank you very much.
Thank you very much, Claudio. I think there is time for one quick question from the audience. Any question? If there is not, I would have one quick question, and it's regarding the next step of your study. Do you plan to, for example, perform new genomic analysis based on SNP data, for instance, to include the adaptive?
We can, we can try, especially now we are working not with this species, but we try to analyze with the rat sequence, and just to see the putative alleles for the, for example, reproduction, or for the adaptative yes, potential of for example, for the temperature adaptations, and other strategies. But as I mentioned before, it's very difficult to do that because it's a tetraploid species. So we must see and yes, yeah.
Thank you very much, Claudio.
Thank you
for your intervention. I think we can go directly to the next talk. She will be held by Nadia Mucci from the Italian Institute for the Environmental Protection and Research, ISPRA. Thank you, Nadia. And the title of the talk is focusing on wildlife, the role of multidisciplinary European funded projects. The floor is yours, Nadia. Thank you.
Good morning to everybody. Thanks for introducing me. And today I'll speak about the results of 3 European funded projects and which my institute was involved with. And there's further evidence of the utility of this project for the conservation and the management of the species. And we choose 3 life projects dealing with the conservation of 3 species, that the wolf, the Italian grey partridge, and the Mediterranean trout. Very different species, but that are similarly treated by the hybridization with allochthonid individuals that are released into the wild. The free-ranging dog for the wolves, the allochthonous European population for the Italian grey partridge, and the Atlantic trout for the Mediterranean trout. Before starting to speak about the project, only 2 words about the hybridization and integration. Of course, this talk will continue the presentation of Claudio before, because we continue to speak about hybridization. And this definition, only to understand the definition that I'm going to use in the next slides. When allochthonous individuals are released into the wild, they can cross with the natural populations, and so they can generate the hybrids. If the event is sporadic, the hybridization event is sporadic, we can expect that the alien DNA will be diluted in the next generations. And if we found an individual with some part of the genome retained, it could be an evidence of hybridization, but a non-recent hybridization, and so we call an introgressed individual. And of course, the worst situation when then the hybridization, the number of hybridization events is high, so the risk at the total admixture, and so the loss of the genetic pool. What can do research and management? Of course, the best thing to avoid the releasing of allochthonous individual into the wild, but we know that it's not possible. In many cases, it's accidental, and so we can think to avoid in this manner. But research and management can help in minimizing the effect or hampering the widening of hybridization. And so the next slides, the next project that I'm going to present you, tried to hamper and reduce the effect of the hybridization in these 3 species. First of all, the wolf, the project Life Miracle. It's a project that ended 2 years ago. We were not involved as partners, but we were demanded to carry out the analysis. And as you probably know, nowadays the Italian population of wolves are not longer on the verge of the extinctions as it was at the middle of the past century. But the individuals are threatened by several factors, conflicts with humans, poaching, high mortality in examples on the roads, and hybridization with the dog. And this project focuses on minimizing the effect of the canine stray animals on the wolf conservation. This project was carried out in 2 Apennine areas. an area in central Italy, the national park Gran Sasso and Monti della Laga. I don't know which is the right translation, the English translation, but as an area in central Italy where the species never disappeared. As an historic in an area is an area of historical presence of the wolf. And the second area is further north, and is a region, mountain region, a national park between Tuscany and Emilia. In this area, the species went extinct, and the area was recognized at the end of the past century. And this is simply the workshop of the project and how it was decided to minimize the effect of hybridization. And of course, I'm going to speak about only the genetic aspect of this project. First of all, it was important to define which area were interested by hybridization. And so it was carried out a non-invasive sampling, so analysis of hares and scats. And the genetic analysis, you can see the protocol on the right side of the slide, allowed us to define areas in which there was the presence of wolf or hybrids. Of course, also of dogs, because in some cases the scats belonging to ranging free-ranging dogs. And when we found areas in which there was the presence of hybridization, we carried out invasive samples. So animals were captured and were analyzed. And the analysis, the genetic analysis, allowed us to assign to a wolf or to an hybrid. When an hybrid was found, it was released, but before it was sterilized, so to avoid the diffusion of hybrids. The protocol allowed us to identify the single individuals and so also some loci for the maternal and paternal lineages. Claudio Ferrari has spoken before about this, the mitochondrial lineages, and also a gene, the key locus that allow to detect the black color that it is a sign of hybridization with the dog. These are the results. The percentage of hybridization is very similar in the two areas, around 10% and 14% hybrids, and 6.5% at 3.5% of introgressed individuals. So in the areas in which we found this hybrid, animals were captured, and were captured 25 animals in the central litter and 17 captured individuals in the second park. And we found 8 hybrid, respectively 8 and 7 hybrid, and 15 and 9 introgressed. I would like to highlight that one individual that was captured was evidently morphological, phenotypically hybrid. But the genetic analysis recognized it as a pure individual. For this explanation, I can use the slide before Claudio Ferrari about the dilution, about the different in color in his slide, because this effect could be the effect of the dilution, and of course of the power of the marker panel that have been used, but in this condition of elution of the dog genome. However, the genetic analysis allowed us to reconstruct the family packs. And so we were able to identify as an hybrid because the genetic part, the family analysis allowed to assign to a wolf per hybrid breeding pair. So we were able to assign also with the genetic analysis to an hybrid. Since in this project the conclusion may be very similar, I prefer to wait the end presentation and speak about the conclusion and the future perspective for all the three species. So I'll start immediately to speak about the second project. And the project is the Life Paradox is ongoing, and it regards the recovery, introduction, and conservation of individuals ascribable to native Italian breed patronage. Ascribable, it's a strange definition. But why? Because the situation of conservation of the Italian grey patronage is critical. I can say it's dramatic. Because the Italian populations went extinct. Many of the Italian population went extinct because of the strong hunting pressure. And the only few remnant populations were polluted by the releasing of allochthon individuals from other European populations. So we can say that the gene pool of the Italian great patronage is extinct and the wild. And the only information that it could obtain about the gene pool derived from the analysis of museum samples, so not more into the wild. So several years ago, before the starting of the project, we analyzed several museum samples from different regions of Italy. And we found that in this individual, we analyzed it with analyzed the short region of the mitochondrial DNA control region, and we verified that the haplotypes that we found in the Italian population were never retrieved in the other European populations, and moreover that this haplotype clustered in a single group, in a single monophyletic group. So we could consider that these haplotypes could be a characteristic distinguishing the Italian from the other European populations. And we were able to find these haplotypes in breeding center, of course, local and small breeding center that never stop to rear the ancient and the historical strains, and that never mixed with other individuals. So we considered that these animals could represent something that could be used to recolonize the historical area of presence of the species where the species disappeared. And this is the focus of the project. As before, I simply show the main work of the project, in which the further sampling of animals, of captive breeding animals, are analyzed, trying to verify the presence of the historical and the Italian haplotypes. Then the animals are used to form breeding pairs and for the production of eggs. Eggs are hatched in incubators, so not naturally, and the chick are used to recolonize to recolonize an historical area of presence of the species that is in the Po Valley, is in a protected area in the Po Valley. Before and after the releasing of the animals, we perform genetic analysis before to verify the genetic variability, and so we use 8 microsatellite loci, and so from a part of individuals that are released, and after the genetic analysis from the feather collected on the ground to verify the genetic analysis. And so this we went to verify the trend of the genetic variability during the whole project. And these are the number until now, more than 50,000 eggs and almost 5,000 analyzed samples. The releasing started only 2 years ago, and until now have been released almost 15,000 individuals in this region. As before, we can. There are several considerations that could be done, and several considerations also on the future perspective. But as before, we'll discuss at the end of the presentation. And so I move to the last project that regards the genetic characterization of Mediterranean brown trout population. The life is stream life and is ongoing, and the issue is that many wild population of brown trout went extinct in the wild because of the releasing of the Atlantic trout for fishing purposes. But we know that a few thousand individuals are left in the fragmented and the isolated small water basins. And so we thought that these water basins could be represent a source of spawners to be used for recolonizing the area in which the species went extinct. In the slide, you can see the 6 pilot area in which we started this project. Of course, the area interested in the area that regards mountain areas, so Monte Marcello Magra, Foreste Casentinesi, Monti Sibilline, Maiella, Pollino, and Forestas. So as before, here is a simply workflow of the project. And there was a first part. The first part of the project tried to record the areas in which there was the presence of the Mediterranean trout. But in this step, we try to categorize the area of study. And so we call it bad conservation status area, the areas in which we found the massive presence of Atlantic trout, and it is in red. And we call good conservation status area the areas in which we found the massive presence of the Mediterranean trout. And in yellow, it is an intermediate conservation area, because in these areas we found both the species. So the red areas are to be eradicated. All the trout should be removed. And the good conservation status are the area source for spawners to be translocated for the reproduction. In the intermediate conservation status has been foreseen the selective fishing, so the fishing and the remotion of the Atlantic individuals only. And the second steps regards the spawner selection. So animals found in the green areas are translocated in the hatcheries where they are reproduced, and the newborn, the fish, are then moved to the areas, of course, of the parks that have been do the eradication. In the area in which it was performed, also the selective fishing. So this is here is the results of the analysis. As you can see from the color, we found in 3 parks one or more than one areas in which there was a massive presence of the Mediterranean trout, and so it was possible to start immediately the reproduction and then the reintroduction. In 3 parks, we were not able to find pure populations, but we found pure stocks in hatchery stocks in local hatchery that reared ancient and local strain. So we can continue the selection and the translocation starting from these hatchery stocks. These are the results, and now we can do the conclusions about this project. Of course, it was at the presentation of the working flow and of some results, but I think that it's evident how this project and the possibility to be half-funded project for the conservation of the species, and we were able to act also in situation where when the hybridization is also widespread. And European funded project allowed us not only to obtain information, but also to act, to hampering and to minimizing the effect of hybridization. And this project are important because also of their duration, because the researchers are able to verify the fact of their action also during the project. And what could be the future perspective? Of course, when we speak about future perspective, we think about genomics, but the advantage of genomic, we know it's not so far, it's now. And in some spaces, there is the application of the genomics, the genomic approach, an example on the wolf, an example on the trout. And we think that genomics could be an instrument also to hamper and also to track the hybridization, so to take action to avoid it. An example, it could be useful to track any genetic variants that can be retained in introgressing individual. Maybe there is a function, maybe there is a function that gives advantage. And it would be very interesting also for museum species to verify the presence of genetic variant in endemic species and verify if this variant remain also after the hybridization, or maybe when the climate changes. And of course, it is also useful, the application of landscapes of spatial genomics because we can analyze the genome, but also an example to track the movements of the genomes in terms of alien species or hybrids. And so as before, not only to obtain the information, but also to be able to act immediately. An example, by hampering the diffusion of alien species or of hybrids. And at the end, I want to thank, of course, all the partners that participated into this project. They are a lot. And of course, all the people that worked with us. And I want to thank you for your attention.
Thank you very much, Nadia, for your intervention, your nice overview of these European projects. So I think we have a few minutes to ask some questions from the audience, if there are. Yes, there is one.
Thank you very much. It's a very important work for the Italian wildlife. I was going to ask, did you check for any relationship between genetics and like behavior in wolf or phenotype in trout?
Not in this project, but we have carried out several studies in which we verified a link about morphological characters and genes, so identify outliers in a SNP project. In the trout, we have not yet started with the genomics. And in the Pardix, we have started a collaboration with the French partners of the project and tried to verify, to correlate the phenotypes of the great alien partridge with the genome, with the same variants of the genome. Of course, the wolf is the most studied species, and so there are more evidence and yes, more information about morphological characters and the genome. And in this period, there is a PhD that is trying to correlate the comportamental, the behavioral aspect with genome in wolf, in dogs, and hybrids.
Any other question? So if there is not, I think we thank you a lot, Nadia. So next presentation will be held by Professor Claudio Cioffi about the Life Urca Proemis project from the Department of Biology, University of Florence. Claudio will tell us about how to mitigate the decline of the European pond turtle in Italy and Slovenia. Thank you. The floor is yours.
Thank you very much for the organizations and for Lisha to inviting me here. I'm really glad to see how pervasive now the genetic tools are and are actually introgressing to the life projects as well, which is a quite good thing, I have to say, so that they can actually help conservation and management decision makers, basically, which is for genetics and genomics has been a problem for years. Of course, even if now there are European projects that are actually trying to bridge the gap between the data that are produced by genomic studies to actual to the actual decision-making process. I'll be brief because this project, unlike the other that we just saw, has not started yet. In fact, it is going to start on the 1st of October. So I'm just going to give you a brief overview of what the Life Urca Proemis, Urgent Conservation Actions Proemis Orbicularis in Italy and Slovenia deals with. The main objective of this project is to mitigate the negative trends and improve the conservation status of both the Italian and the Slovenian population of the European pond turtle, Emys orbicularis, while, and this is particular for my department, while maintaining the extant intraspecific genetic diversity. It is a live project, of course, so it has a number of practical conservation actions. The first of which is the restoration and or the reduction of the level of encroachment of Emys orbicularis natural habitats. So this includes marshes, ponds, bogs, rivers, wetlands in general. And this will be done by establishment of new habitats, so building new ponds, by rehabilitating, extending and diversifying wetlands, and by the creation of semi-natural refuges, as well as construction of barriers, fences to prevent intrusions of predators and ungulates, and construction of hydraulic works for water control. So these are very, say, practical interventions before the project will start dealing with the eradication of invasive aliens, like the North American Trachemys species, which are, as you all know, basically are pose a threat in terms of competition for natural habitats with our shy Emys orbicularis. And at the same time, the restoration and or the amelioration of selected rescue and captive breeding centers for Emys orbicularis that are present across the Italian territories. After this reintroduction of captive Emys orbicularis into the wild, of course, following proper health check, so veterinary work and genetic assessment of population of origin. And then there are a number of following complementary actions, of which very important one is the development of a master plan, which will include guidelines for management and conservation of the species, of course, training of operators and technical staff, and because we are working together with two nature reserves from Slovenia, the promotion of cross-border management models in cooperation with these, in particular with the Ljubljana Marshes Nature Park and the Sečović Salina Nature Park, and also other Slovenian institutions. Then the implementation of a long-term monitoring plan for the species, and of course, community awareness work on the protection of Emys orbicularis, and particularly the threats posed by the invasive alien species, which is very hard to make people to understand, not to release these species in the wild when they grow up, basically. And then long-term replication of good practices. So this project is a nationwide project. It involves 55 Italian sites and 3 Slovenian Natura 2000 sites, located in regional parks, state nature reserves, regional nature reserves, and other protected areas. And of these, 4 wetlands are part of the Ramsar Convention, and 2 of them are part of the UNESCO MAB reserves. Here, I don't see if you can see in these maps, but basically these red lines here, and so on are all the sites where the project will take places. So in continental Italy and Sicily, but not in Sardinia, at least for this project here. These are the partners involved. We have 3 NGOs. Basically, the coordinator is the WWF Italy. Then we have the Aquarium Genoa and the Centro Studi Bionaturalistici, which is a spin-off, basically, of the University of Genoa. 2 governmental organizations from Italy and 2 governmental organizations from Slovenia, and 3 academic institutions, the University of Florence, University of Pisa, and University of Modena and Reggio Emilia. As I was mentioning before, what we at the University of Florence are particularly interested about in this live project is the point number 4 that I've shown before. So the reintroduction of captive Emys orbicularis into the wild. And this is included in the work package for genetic-based determination of population of origin and kinship of the European pond turtles for breeding and restocking. This work package basically is based on relatively, say, simple genetic assay using standard markers, although we are expanding these to whole genomic approach. So the aim is basically to profiling and then determining the population of origin of captive individuals. And this is done because we have already a database on the genetic structure of the main natural population through the Italian territories. So basically this implies to do an assignment test of the individuals that we have in captivity with the genetic structure of the natural populations. Then selection of best mating pairs among parents, among basically the individuals that are kept in the captive facility. And this is based on kinship values, of course, in order to maximize then the genetic variation of the offspring. And because, as you know, in reptiles in particular, in reptiles and in turtles, and tortoises in particular, the females can retain the semen from previous mating events. We will have also to profile the hatchlings born in captivity, although we know which parents they come from, basically to confirm the genetic origin or assess possible fertilization from semen of previous mating events retained in the female oviducts. So these are the, let's say, main. I'm just presenting this part rather than all the rest, because this is the part that we are involved with. Just describing what this part includes, of course, as I was saying, the assignment of captive individuals to the population of origin, and the population of origin has been described by sampling these individuals through the Italian territories. So capturing them and collecting samples invasively, in this case, blood samples that, as you know, can be maintained in the field using proper lysis buffers. And then using standard molecular markers from the mitochondrial DNA that we have all seen during this morning presentation, and nuclear DNA markers. In particular, with the mitochondrial DNA markers, we have been doing a first screening using the control region, which we found to be a bit more variable at the intraspecific levels, at least for the European pond turtles. And you can see from this first distribution of haplotypes, the numbers and the colors here basically are the same. That means we have about 20 different haplotypes in the Italian territories. You can see the classical subdivision from the east and western coast of Italy, and also the larger genetic diversity that we have in the south part of Italy that correspond to the genetic diversity retained during the glacial refugia. The distinctiveness of the Sicilian population compared to the other part of Italy, So, and was the reason why the Emys orbicularis in Sicily has been elevated, say, to a different species status for now, it's called Emys trinacris. Okay, so from this map, one can see that it is easy to tell whether a captive individuals could belong to a population from the west or east coast of Italy, but it will be quite difficult to understand whether a captive individual belong, for example, to the Ticino valleys or to the delta or to the Po or to the delta of the Po River. So one way of going further is to expand from the from using just a single section of the mitochondrial DNA, so in this case the control region, to the entire mitochondrial molecules, something that we have been doing with other species, with the marine sea turtles, for example, Caretta caretta. It works pretty well if we do a whole genome sequencing approach, low coverage whole genome sequencing approach, basically 1-2 pair for the nuclear genome, which results in a very high coverage, 200-300 pair of the mitochondrial genome in this case. So this is a way of, say, refining the genetic structure of the natural population, and the other is to also complement these to the nuclear markers that so far we have been using the standard microsatellite loci that we characterized for Emys orbicularis, and of course these are it gives an additional level of resolution when we have to assign single individuals to the population of origin. And again, for these, for the nuclear genome, this is an example of an assignment test where in green you see the samples, the individuals, the captive individuals that have been assigned with a relatively high probability to a particular natural populations in the north, here in the north part of Italy. So as for the mitochondrial DNA, even for the nuclear DNA, to have a larger resolution, as we have already seen during this morning presentation, is to go to next level, so to do single nuclear polymorphism analysis that nowadays can be done using a RAT-seq approach, particularly if the samples are not really good, but if the samples are good, one can, and high quality DNA extraction, one can go directly to a whole genome sequencing approach. And so and the resequencing if a reference genome is available. And in our case, we have developed the first chromosome level reference genome for Emys orbicularis, and so it will be easier to do a resequencing on the reference genome to find the SNPs to complement to the microsatellite analysis that we are currently doing. The microsats analysis for now is providing good results for kinship analysis and paternity tests. So this might be enough, at least for the first part of the project, where we have to determine the best mating pairs and also to check that the hatchlings really come from the pairs that we have chosen for mating. These among the different loci that we are using, these are only 3, 6, 9 loci. We are using about 14. These square here are shifted basically to the left, but they were meant to be on this locus called Emys 3, that is one of the loci that is able to is a diagnostic to assess multiple paternity in this species, and therefore to see whether a female has a hatchling that belong to the male that we have chosen as best mate, or it also retain semen from previous mating events. And in some cases difficult because these species has not a very large output. Sometimes you have only a couple of eggs, and in this case you can assess, of course, whether the female is whether we have an event of multiple paternity or not. And that's it. So I think we have time also to go to lunch.
Thank you,
The time.
Claudio, for your presentation. Any question from the audience? So if there is not, I would have one. So more a curiosity than a question. You show us that there is this kind of genetic isolation in the south of Italy regarding based on the mitochondrial DNA, right? So my question is, there are data already if this situation is also reflecting in the nuclear by using nuclear markers, or it's just like the same?
And we haven't tested that with the nuclear markers, but more than I think more than genetic isolation is more the aftermath of the classical glacial refusal phenomena that we see in other species as well. In Italy, in which the south you have this greater genetic diversity, and because of these relatively recent colonization event, then to the north, of course, the northern population show only a fraction of the genetic diversity that you find in the south. Yeah. And also because that, I mean, the lower genetic diversity that you find in the south has been basically halted then by the habitat fragmentation and encroachment.
Which in fact, another question, my side, do you have an idea about the conservation status of the species in Italy already, or we don't have any information, or?
Well, there are basic informations. Part of the, probably I didn't show it, but part of the project is also to assess to reassess the population, say, estimates of the species in main areas in the 55 different sites that we have. Of course, the conservation status is basically, say, declining because this species is very linked to the wet habitats. So there are some populations that are found at high altitudes in the central part of Italy, in Calabria, for instance, so in Basilicata, up to more than 1,000 meters, and they're very isolated. So they're just ponds that are isolated in woods. And others, like in Tuscany or through the northern Italy, are all linked to marshlands, basically. And here, particularly here in Piacenza, we sampled Emys orbicularis around 5 or 6 years ago, and those populations are now gone, basically, because of they destroy the habitat.
Also, climate change, I imagine, will impact then changing patterns of precipitation or aridity.
Sorry, in Piacenza, Cremona. Sorry, not in Piacenza, in Cremona.
It's very near from here. So, I mean, climate change is expected to impact a lot this, let's say, the extirpation risks.
Well, it is mainly because of the temperature sex determination that you have in turtles.
Sure.
I mean, we already have in some, not in pond turtles, but in sea turtles, instances in which you have a clutch that is composed by all females, for instance, and that, of course, is also a main threat to the extinction of a species, basically.
Yeah, sure. Thank you. I think there is one other question from Claudio.
Thank you very much for your presentation. I'm very curious about the regarding the selection of the female selection. If I understand quite well, is the female they can select the male sperm, right? In the for example reproduction in the reproduction season, so they can collect more for example.
Yeah.
Okay. So you
so
can
if it's whether it's a female they select,
yeah,
they choose the male or that's,
exactly.
that's
the first question is this one: if you want to study even the behavior of the male in terms of, for example, the female, and how for example, the behavior can reflect the selection of the sperm, if they can do that.
Yes, you can. I think there are a couple of studies published on this. But basically the aim here is to increase basically the genetic variability of the hatchlings. So, even if, okay, those are, there have been and could be also side studies, but our main interest is to find individuals that are compatible from a genetic point of view. Compatible, I'm saying they are not, that their kinship level is not too high. And of course, you can't, force individuals to mate. So at least for this species, you will have to put maybe one female and more than one male, and then see, of course, more than one male and both males, or if you choose three males, they will have to be all compatible from a genetic point of view to the female. Not the parentage level shouldn't be too high, basically. And then you can see from the hatchlings which one has succeeded, or if two of them, or say more, or all of them have succeeded in mating with the females, basically. Yeah. It's as all life project, this is as well 5 years. Oh no, some life are even short. This is a 5 years project, and of course you had to allow for the hatchlings to grow, to be also to be reintroduced, because if you reintroduce the hatchling when they are too small, they will be predated. So at least they will have to have at least 3 years. They have to be at least 3 years old to be of a size that with which you can minimize the possibility of them being predated by raptors, for example, or others, or rats or foxes and so on.
Any other question? So I think if there is not, we can thank a lot Professor Claudio Cioffi. Thank you very much for your intervention. Thanks.